Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi.
First of all, let us send our praise to Allah subhanahu wa ta'ala for all the bless, all the mercy, and all the joy that had given to us, so that today we could gather together here in the fourth series of the S2FiMaS. Ladies and gentlemen, this is a wonderful day. I am Diahruri Sanjay-Sari. I feel very honored to be your MC today for the seminar series. And the S2FiMaS is organized by Fisheries and Marine Science faculty, Jenderal Sudirman University. And the seminar series will held for 5 series, with the first series started since 19th of November 2020. And the 2020 round of the seminar will be end on the 17th of December 2020. There are 12 expert speakers and 100 participants join this online seminar series. And hopefully, it will be valuable experience and incredible opportunity during this pandemic for everyone here. We are not only attending, but also sharing in-depth knowledge and even connecting and reconnecting with colleagues around Indonesia and around the globe. Hope this seminar will be the memorable one. So ladies and gentlemen, we have to apologize that due to the time difference of the invited speaker, we have to switch the agenda. So if in the previous seminar series, we have the expert speaker session in the beginning and following with the parallel session afterward, but today we're going to switch. The parallel session will be held at the beginning. So we hope all the presenters in the parallel sessions are ready to present their presentation today. Distinguished guests, ladies and gentlemen, we now come to the parallel session. This session will be led by our moderator. But before we start, allow me to read his curriculum vitae. Organizer or committee, can you probably show our moderator CV today? So he is a lecturer in Fisheries and Marine Science faculty, especially in aquaculture department, Jenderal Sudirman University, and he has expertise in fish nutrition. I hope everyone can see his CV. Yes, this is Mr. Ren Fitriadi, MP. So let us give him a warm welcome. Mr. Ren Fitriadi, MP, the time is yours.
And the last presenter is Miss Titin Yulianti with the title Characteristic of the Sea Level Anomaly During El Nino and La Nina in Southern Water of Java. Each speaker has 10 minutes for to present their paper and continue for 20 minutes for discussion section. Please, Mr. Amron, you have 10 minutes for the presentation. Mr. Amron?
Yeah, okay, Mr. Ren, thank you. Hello.
Can you hear me, Mr. Amron?
Yes, I can hear you. Hello. My sound is clear. Hello.
Apparently, some of the committees try to find the problem. Yeah, the moderator cannot hear it.
Is my sound is clear?
Yes, sir.
Okay. I can start now. Hello. Hello. Can I start now?
I think yes, Pak Amron.
Can I start now? Okay. Thank you. Can I share my presentation? This is still closed. Please, operator, please allow me to share my presentation. Because host disable me. Hello. Please host disable me to share my presentation. Hello host. Please allow me to share my presentation.
Yes, wait a moment. We still managed to make you a co-host. Wait a second.
Yeah, I try to, yes. Yeah, hello, already appear. Hello.
Yes, all good.
Okay,
Because
thank you very much. Yeah, this is my time. Hello.
Yeah, can you show us a slideshow, please?
This already, I already saw this presentation.
Yes,
Already appear, yeah.
thank you.
Yeah. Thank you very much for the moderator for the committee. Assalamualaikum warahmatullahi wabarakatuh. Good morning. Sorry, is my voice is clear?
Yes,
Sorry.
please, sir.
Yeah. Thank you. I would like to present our research entitled Effects of the Water Salinity on Sound Production of the Spiny Lobster Panolirus homarus cultivated in the laboratory scale. Uh, this is my uh, article with my college, Sudibya, Isti Sulistio, and me Amron, Hidayat, and Wijayanto. This is the reason why this research is important to be conducted. Uh, the high demand for this commodity, Panulirus homarus, uh, both from the domestic or and the export commodity, make the fishermen to increase the exploitation of the species. The impact of this condition is uh, overfishing is a cure to this species. So we need one effort to manage this situation, and the one of effort is cultivation. However, uh, for successfully for the cultivation, many factors that impacted this activity, and in this research, we focus to the water salinity, water quality, especially the salinity impact to the sound production. Because in the uh, marine habitat, we are difficult to investigate the impact of the water salinity to the habit, the the performance behavior of this species. So we need one and uh, one method to investigate this behavior. We call the sound production. We have investigate the sound production to uh, study the effect of the water salinity to the behavior of the lobster. This is our method. Uh, we are conducted the observation method in the laboratory. We place two lobster, one lobster in one aquarium and the other lobster in the other aquarium. Uh, we we try to record the sound behavior from the lobster which using the hydrophone and connected to the uh, personal computer. And the other method, we apply the CCTV camera to record the performance behavior of the lobster. Both the sound recorded and video recorded, we synchronized. And the aquarium, the salinity was declined for 2 ppt per day from 32 to 14 ppt. This is the sound production of the lobster. There are three kind of sound that produced by the lobster. Is the first is squeak sound, and the second is slow rattle, and the third is popping. Uh, all of the sound from the lobster is produced by the a couple of the organ of lobster. We call uh, this organ is file and plectrum. From the slip and stick mechanism, from this from the couple of this organ, will produce squeak sound and uh, and popping and slow rattle. The difference, the difference of the sound is impacted by the higher of quantity of the stick and slip mechanism and the number of this mechanism. Squeak sound produce the number of sound pulse. In the figure, we can see so many sound pulse. And then this figure B show the frequency of this sound, and figure C sound show that the pulse duration of the sound. We can compare to the slow rattle sound. Slow rattle sound consists of 1, 2, or 3 sound pulse. We can see here, and the frequency is not different with the first sound, and the same case is the sound the duration of sound pulse is same with the first sound. And the difference with slow rattle, the difference between slow rattle and squeak sound is we can differentiate by the number of pulse duration, number of pulse sound, and the intensity of the sound. But the frequency and the pulse duration is similar. And the last sound is we call the popping sound. We can see here only one pulse sound in this sound type, and the frequency is not different with the other sound. In the same case, it's not different with the pulse duration. Popping sound is produced by the lobster as representation to the defense behavior. And slow rattle sound is produced when the lobster is what is it? Feeding behavior. And the first squeak sound is produced when the lobster is spawning behavior. This is the difference of the each of sound. And what is the impact of the salinity to the sound productivity? We can see here. This is described to what is the impact of the salinity to sound productivity. This blue line interpret that if the salinity is decreased until 20 ppt, the productivity is decreased too. And after 20, the sound production is increased. In the same case for the lobster B in the red line, the same situation, if the water salinity is decreased until 20 ppt, the number of sound that produced by the lobster is decreased too. But it's the same if the increase of the decrease of the salinity, water salinity, the number of sound is increased. It means that the the minimum of salinity that will be adapted by the lobster is 20 ppt. It is the optimum salinity of the lobster from 20 until 40. This is the result of our research. Thank you very much for this presentation. We back the time to the moderator. Thank you very much. Assalamualaikum warahmatullahi wabarakatuh.
Okay. First time I will like to share screen.
Okay.
Is my sounds clearly? And my PPT also already show up?
Yeah.
Okay, thank you.
Okay.
Assalamualaikum warahmatullahi wabarakatuh. Good morning, ladies and gentlemen. My name is Camelia from Institute of Marine Research and Observation, Ministry of Marine Affairs and Fisheries. Today, I will share our research work. This is a preliminary research with the topic is the response of tuna larvae to ocean acidification with case study in the Gulf of Bone in Indonesia. Nowadays, there is a growing concern on ocean acidification. It's defined as decrease in ocean pH due to the increasing of atmospheric carbon dioxide that absorbed by ocean. This ocean acidification may have different level of risk on many marine organisms. Previous work suggests that ocean acidification revealed consistent in reduction in calcification and growth and development of a range of calcified organisms such as coral and sea cells. But meanwhile, some taxa may be predictably more resilient or may benefit from ocean acidification, such as microalgae, diatom, and fish, including tuna. This tuna is the most valuable fisheries species in the world in terms of catch weight. It is a key component of pelagic ecosystem that provide a significant contribution to the economic of many Pacific island countries and territories. A few research has been done to investigate the effects of ocean acidification on the species of tuna. The research work by Scully show that the genotype of 4 main species of tuna has an effect on response of eggs on larvae to ocean acidification. Another research by Portner and Bromhead revealed that ocean acidification could have potential impact on the eggs and larvae of yellowfin tuna, but it provides some inconsistency in results. So it was relatively clear that the ocean acidification effect on larvae were not evident until the partial pressure of carbon dioxide level reached above 1,500 microatmosphere. As we know that Indonesia is one of the main tuna producers that contributes more than 600,000 metric tons of tuna per year. Most of tuna fisheries in Indonesia are operated in the eastern region. This area is one of the most potentially great tuna fishing grounds in Indonesia waters. However, there is a critical gap of information about the effect of ocean acidification on tuna, particularly in the Gulf of Bone in Indonesia. This is our study area in Gulf of Bone in South Sulawesi province that belongs to the Indonesian Fisheries Management Area 713. In this research work for the analysis, we use the pH and partial pressure of carbon dioxide data series from SIMEMS model product and fish data from Delhi Indeso tuna population model output. In the analysis, we analysis pH and partial pressure of carbon dioxide data from 2010 to 2019. The result analysis show that during 10 years observation, the variability of pH tend to decrease in the red line, while the partial pressure of carbon dioxide tend to increase. The highest monthly data series was observed in July 2011 and the lowest pH was in December 2019. And then we continue to statistical analysis. It was found that the correlation of pH and partial pressure of carbon dioxide was strong with R value of 0.96. In comparing seasonal variability, generally pH in the Gulf of Borneo was lower during northwest monsoon on figure A compared to the southeast monsoon in figure B. This result coincides with the partial pressure carbon dioxide data, which show higher concentration on northwest monsoon in figure C compared to the southeast monsoon on figure D. So from this result, we assume that the monsoonal pattern plays significant role in the variation of pH and partial pressure of carbon dioxide of seawater in Indonesian waters. This is the figure of yellowfin tuna larva abundance in Gulf of Borneo. Due to the availability of fish data, we analyzed from January 2014 to April 2017. The analysis showed that the average of yellowfin tuna larva in Gulf of Borneo was 0.4 number per square kilometers. The highest abundance of larvae was found in January 2015, while the lowest was observed in May 2014. During 3 years observation, the yellowfin tuna larva data series in this area were tend to increase. For further analysis, they show that the increase of yellowfin tuna larva in red line, it coincides with the increase of partial pressure of carbon dioxide in figure A and the decrease of pH in figure B. And from the statistical analysis, it showed that there was a very weak correlation between yellowfin tuna larva to both of partial pressure, carbon dioxide, and pH. It is 0.19 and 0.07 respectively. In conclusion, our study indicates that the density of yellowfin tuna larvae tend to increase with the decrease of pH and elevated partial pressure of carbon dioxide, but the correlation was very weak. This does not necessarily indicate that the species will not affected by ocean acidification because the slow and long-term change of pH in seawater may encounter increasingly stressful conditions in the coming decades. And then bring us to the end of my presentation. Thank you so much for your attention. Wassalamualaikum warahmatullahi wabarakatuh.
We are going to the third presenter. Please, Mr. Victor David Gultom. You have 10 minutes for the presentation. Mr. Victor.
Thank you. Thank you.
Okay.
Can you see my presentation?
Yes. The
Yes. Can I start my presentation?
The time is yours.
Okay, thank you very much. Thank you for having me. My name is Victor David Gultom. I am a lecturer in Universitas Asia Negara Indonesia in Jakarta Selatan. In this conference, uh, I will present the result of my experiment when I was working as a researcher in South Korea in a flounder farm. I conducted traditional sorting of unwanted physical traits in juvenile Japanese flounder. So first of all, juvenile flounder is the most important aquaculture species in Korea. Japanese flounder is the left-eyed flounders, which means that they are part of flatfish group because they can lay flat at the bottom of the ocean, and then they have a common name in Japanese flounder, olive flounder, or bastard halibut, and also they have local names such as nupchi in Korea or hirame in Japan or hea in China. So this flounder is mainly used for consumption as a raw food, sashimi. You can see there are some Koreans usually eat flounder or raw food as most important occasions such as birthday or wedding. So, and then there are benefit of consuming flounder. The meat can build up strength, they can revive the energy, and then also increase the appetite. And other than for raw food, then the flounder can be cooked as soup or stew. It is best for people with diabetes, liver disease, or currently recovering from illness. So why they are called flatfish? Because at a certain point of their life stage, they undergo metamorphosis, and at this stage, the flatfish there are two kind of flatfish: the left-eyed flatfish and the right-eyed flatfish. In this case of flounder, they are mostly left-eyed flatfish, which means that all the eyes gather at the left side. Uh, at at the 20-day post-hatching, these fish undergo metamorphosis when the right eyes move to the left eyes, and then during the metamorphosis, they gradually swim closer to the bottom, and then they lay on their side and then live at the bottom as demersal fish. So the metamorphosis and the attaching to the bottom is are the most important life stage in flounder, because failure in metamorphosis and attachment will result in abnormalities. And then I will explain about the aquaculture production of Japanese flounder. In terms of quantity, they are twice bigger than the Korean rockfish, other finfish aquaculture production. And then the aquaculture production is much higher compared to capture production of Japanese flounder. That is 4.7 thousand tons. However, the total aquaculture production, including mollusk and aquatic plants, is more than 2.3 million tons. Compared to Japanese flounder, the number are very small. This is because the mollusk and aquatic plants production is much higher than flatfish production. But however, the price of the ratio of per kilogram and then the value is much higher in Japanese flounder. First of all, I would like to explain about the production of juvenile flounder in Korea. First, juvenile production is already well established in Korea, and then government already supplied manual that is continuously updated every 10 years or 5 years. Usually the spawning temperature is optimum temperature is 17 centigrade, and then after 5 to 7 days after spawning, the usually the spawning is conducted at breeding center owned by government, and then the government will transfer the fertilized egg to farm across South Korea. And then to transfer the fertilized egg, it took 15 to 20 hours. And then they will hatch at a temperature between 14 to 17, mostly 17 centigrade. And then after more than 2 days in 15 centigrade. And then after hatching, the farmer usually increase the temperature from 17 to 16 centigrade with a rate of 0.5 centigrade per day. And then they will start feeding rotifor, artemia, and then starter feed like the powder feed. The metamorphosis period is the most important period. It happened at 20 to 25 day post hatching. At this moment, they start swimming close to the bottom, and then when the fish, most of the fish attached to the bottom, the farmer usually stop feeding the artemia, and then the remaining fish, the one that still swimming at the surface, they disperse to the fish to the other tank. The end of metamorphosis stage is occurred around 30 to 40 day post hatching, and then after end of the metamorphosis, the farmer can start grading. Grading means that they only separate the fish based on the size, not by the shape or by the appearance. They use first they use 5.5 millimeter grader. These are the size of the mesh. It means it's not their length, but it is their body depth. And then after grading, around 67 day post hatching, they can start sorting. It means they grade the fish first, and then after grading the larger fish, they will sort the fish based on their external appearance. Some abnormal and unwanted physical traits like malpigmentation or abnormal shape will be discarded. And then after sorting, they can start selling the fish at the age of 70 day post hatching. These are the instruments used for sorting process. First, they transfer the fish to the grading container, and then the smaller fish will escape to the tank while the larger fish are trapped inside. And then the fish inside the grader can be transferred to sorting bench or transferred to temporary net. So in this sorting bench, the person who will sort the fish based on the appearance, some abnormal appearance or unusual shape, they will ruthlessly discard it, just like this. And then the normal fish is transferred using pump to another tank. And then there are problems encountered during juvenile production. Mainly the problem is abnormal physical traits. There are high incidence of abnormalities, sometimes close to 20%, and other times close to 55%. These are the abnormal traits such as malpigmentation, skeletal anomalies, incomplete eye migration, and shortening of operculum. And abnormally seriously affect the market value and the production cost. If you imagine that the abnormal rate is over 50%, then the production cost will be much higher. And then of course, there will be nobody who wants to purchase abnormal fish, and then the market value will be reduced. In sorting of unwanted abnormal fish. However, there are a lack of technology to sort abnormal flounder because sorting is done by traditional method, manual sorting using only net, and sorting is conducted by middle-aged women. They are highly experienced and they have been sorting flounder for more than 10 years. They are very accurate and then very fast. They can sort up to 3,000 fish per hour per person. However, there are problems in traditional sorting because the person lack of highly experienced person. The next generation of this after this woman, there are no second generation. And another problem is in the spring there are usually high demand of to sort fish, then require appointment with the, and then also scheduling, and this resulted in sorting delays. And also usually one farm only trust and use and work with one group, and usually didn't work with another group. And then because it's based on experience, there are no standard, and then it is highly subjective. These are the abnormalities mostly found in flounder juveniles. I differentiate based on the body shape. We can see that these, we can recognize immediately the shape, the undulated shape from dorsal to head, and then dwarfism, their size is much smaller than their conspecifics. And also some fish have a slightly rectangular shape. Normal fish usually elliptical in shape. And abnormal pigmentation also commonly found in flounder juvenile. In this case of broad area of malpigmentation, even a small area of malpigmentation is discarded. And at some case, it can reach to 24% or 100% of juvenile reared in aquaculture facility. In order to reduce the malpigmentation, farmer is encouraged to add vitamin A and DHAs during the Artemia enrichment. Another abnormalities are spinal column deformation. We can see there are convoluted spinal cord, and then we can see there's deformed spinal cord at the ocular side. And then there are also these abnormalities in skeleton or head is most commonly encountered. However, it's very difficult to distinguish for novice because usually some minor details can be found after 50 days post hatching. And for example, like this gap in operculum, if novice person cannot distinguish the difference. And then there are also deformation in the upper column, and then the malformed or undeformed upper jaw. You can see the upper jaw is much smaller, much shorter than the lower jaw. And also the malformed upper jaw, and then gap in operculum. Usually operculum deformation is caused by genetics. Conclusion for my experiment: skeletal abnormalities, especially head, are the most commonly found deformities. It usually happen in densely packed hatchery juveniles, and most abnormalities occur and during the metamorphosis stage. And then also the attachment, some larvae late because they grow at later, they late attach to the bottom. So in the bottom space already occupied by other fish, and this result in the malnutrition of late attachment. And success of metamorphosis and attachment is very essential for the survival of flounder larva. These are the completely normal image of juvenile flounder. Yes. Thank you for your attention.
Victor David Gultom, for your great presentation. Now we will have a discussion section. The audience may ask questions by activating the right hand or writing question in the chat column. The audience, any question?
Can I ask question?
Okay, Mrs. Diarudi.
Thank you very much for the opportunity. So first of all, I would like to ask to Mr. Amron. So in your study, you mentioned several sound of the spinal lobsters. There's a click, and I can't really recall what is the other sound. But can you possibly explain how that kind of sound produced, and would it be possible the sound will be the same for other species? Yeah, that was the first question for Mr. Amron. The second question I would like to give to Mr. Victor. It's very interesting research related to the flounder fish. So my question is, if there is a deformities in the skeletal of the fish, would it be reduce the cost? I mean, reduce the price of the flounder fish in the market, or what would be the effect for the economy? Thank you.
Okay.
Thank you.
For your question, please, Mr. Amron, to answer the question. Mr. Amron.
Hello, the sound of moderator is unmute. Yeah. Thank you very much for the question. As commonly, the sound of fish is differentiated by the organ sound reproduction organ and the mechanism of sound production. For this species, the production of the sound is produced by the two, I mean one couple of the organ of the lobster. From this organ, from this couple of organ, can produce the three kind of the sound of lobster, because only one couple of the organ, so the differentiation of this sound is not too different, because this is only produced by the couple of a couple of the organ. If the different mechanism, of course, they can produce the other sound, but this is the only couple of the organ. So the differentiation of this three kind of sound is not different. And how to the other species? If the organ that produce the sound is different, of course they can differentiate of the sound that produced by the species. For the lobster, almost the sound is produced by the couple of this organ is from the plectrum and the file, and one mechanism we call the slip and stick mechanism. So that why the sound that produce all of the lobster only has three kind of the sound: slow rattle, popping, uh, and squeak. This my answer. It means that the differentiation of the sound, the characteristic of sound is uh, delivered by the organ that that produce the sound and the mechanism of sound reproduction. Thank you very much.
Yes, thank you very much for the question. Uh, Actually, in case of deformities in Japanese flounder in Korea uh, accept is usually acceptable. It means close to 5 to 10% of the total production. So in order to eliminate this problem, farmer usually stock more larvae. So for example, when they plan to produce around 500 juveniles in this period, they stock 500,000 plus 100,000. So they can get extra, the total uh, uh, normal fish much higher. And also in case of uh, abnormal larva, usually the breeding is conducted at the specified breeding center owned by government. So they already know the broodstock lineage. So it's very rare to, in my presentation, the abnormality rate can go up to 25 to 50%. But in case of Japanese flounder supplied by breeding center in Korea, it's usually around 5 to less than 10%. But Korean public usually doesn't like the form specimen. They some minute detail on slightly abnormal pigmentation. The that fish will stay in the aquaria for a very long time, and then it will make the price, the market value lower. So usually, farmer conduct sorting every 2 weeks, around 2 weeks. So even they already sort the fish, and then they will sort again the fish within 2 weeks later. That is my answer.
Victor David Gultom. Mrs. Diaruri is Mr. Amron and Miss Victor David Gultom. Answer is clear.
All good. Thank you.
Any question? Okay. Any question in chat room? Any question in chat room from Hendrayana to Mr. Victor, what the benefit of your
Thank you
research
for the
for your country, Indonesia? It is type of fish name in Indonesia.
uh, thank you for the question, Mr. Hendrawan. I searched. I noticed that there is also certain flounder or flatfish species in Indonesia. It is located around the Sumatra, located uh, between Indonesian coastline and also can go to the southern for domestication and for further breeding. Because however, the flounder fish in Indonesia is much smaller than compared to the flounder fish in Korea. The maximum size is 15 centimeter. So perhaps in the future, if the process of sorting and then can be implemented in Indonesian aqua farm. This is my answer. If you have further question.
Okay, any question in chat room? From Mr. Hardoyo to Mr. Amron. There, Dr. Amron, is it possible to ask the fount of aquatic organism to see or make a prediction of the chemical contaminant in the water?
Sorry, moderator is not too clear. So
Okay.
it's difficult to conclude the question.
Amron, there, Dr. Amron, is it possible to ask the son of aquatic organism to see or make a prediction of the chemical contaminant in the water?
Yeah, thank you for the question. Sorry, the sounds of moderator is not too clear, but I can catch the message. Of course, the from the sound characteristic, we can predict the impact of the chemical composition of the water uh, to the species, because one of uh, the objective of uh, the species why they produce the fish is as a response to the environmental change. So if the environmental change, we can impacted the sound production of the fish. Is it relating to the behavior of the fish as an impact of the change of the chemical composition of the waters? This is my question, my answer. Thank you, Pak Artoyo.
After the discussion section, we will continue the presentation of parallel session. Start with the fourth speaker. Please, Miss Mila Santika, you have 10 minutes for the presentation. Miss Mila Santika.
Yes, I'm here.
You have 10 minutes for the presentation.
Okay, thank you. Okay, Assalamualaikum warahmatullahi wabarakatuh. Thank you for having me. Good afternoon, everyone. My name is Mila Santika. I study from University of Jenderal Sudirman, Purwokerto. Today, I will present about my research entitled is Citrus Leaves Simodosia rotundata extract as again Aeromonas hydrophila and Vibrio harvey. Introduction. Among the bacteria infection in fisheries agriculture can be considerable cause to business. The types of bacteria infection can usually affect fish and shrimp. Bacteria-induced disease show like loss of appetite, scar on the surface of the body, hemorrhaging in the gills, abdomen full of fluid, loose skull, loose tail fin. If surgery is performed, there will be swelling. Typically, treatment of bacteria infection are being treated with synthetic antibiotic like doxycycline, enrofloxacin, gentacillin, and ampicillin. Antibiotics are a set of molecules, both natural and synthetic, that have the effect of suppressing or stopping a biochemical process in organism, especially in the process of bacteria infection. The effect of the use of synthetic antibiotic can cause resistance and can affect water pollution. As similar types of citrus common in Indonesia as Cimodocea rotundata. Cimodocea rotundata citrus has several bioactive compounds such as tannin, saponin, and flavonoid. The citrus of Cimodocea rotundata has not been widely used to replace natural source of antibacterial, so that raw material for antibacterial production will continue to be available. Research question. First, what are the bioactive compounds contained in the extract of citrus leaves Cimodocea rotundata? Second, what is the antibacterial potential of leaf extract Cimodocea rotundata at the inhibition zone diameter against bacteria Aeromonas hydrophila and Vibrio harvey? Research purpose. First, to find out the bioactive compound in the citrus leaf extract of Cimodocea rotundata. Second, to find out the antibacterial potential of leaf extract Cimodocea rotundata based on the diameter of the inhibition area of bacteria Aeromonas hydrophila and Vibrio harvey. And then these are a main tool I use. First, rotary evaporator, shaker incubator, paper dish, water bath shaker, and then petri dish. And these are main tool. These are main material use. Cimodocea rotundata, Aeromonas hydrophila, Vibrio harvey, tripsid soy agar media, antibiotic ampicillin, pro-analysis methanol, aquades, water, and then etc. And then sterilization. Tool sterilization. Using incandescent with direct fire to burn the tool on the fire directly. Using dry heat in an oven at 170 degrees Celsius for 1 hour and autoclaving. Material sterilization, autoclave and last for 15 minutes at 121 degrees Celsius. And then these are some research method. First, sampling, and then second, free extraction citrus leaf of Cimodocea rotundata. Extraction of citrus leaf Cimodocea rotundata. Fourth, analysis of phytochemical compound of leaf seagrass extract, Cymodocea rotundata, and then the last, test for the antibacterial activity of extract leaf, Cymodocea rotundata. This is the second method is pre-extraction of Cymodocea rotundata. Cymodocea rotundata sample source spiration of leaf, roots, and rhizome. The sample was washed 5 times using fresh water. The sample was dried, and then the sample was weighed, and then the sample were brined to air at room temperature without exposure to sunlight, and then dry weighed. And then this is the search method is extraction of Cymodocea rotundata leaves. 40 grams of Cymodocea rotundata leaf sample were put into Erlenmeyer. 200 milli of pro-analysis methanol solution was added to the Erlenmeyer, and then the sample was homogenized with an incubator shaker at speed of 120 degree RPM. With 48 hour with a change of methanol solution every 24 hour, and then the filtrate was evaporated using a rotary evaporator at temperature of 64.7 degrees Celsius. The solvent evaporated, and then the sample was concentrated using a water bath shaker, and then the sample is filtered, and the last safe extraction. And then the next method is phytochemical analysis. First, tannin test. A total of 0. grams of Cymodocea rotundata seagrass leaf extract. Add to hot and simmer 5 minutes, and then add 1% FeCl3 reagent. And then if the solution produced is the blackish green, it is positive contain tannin. And then the second flavonoid test, a total of 0.05 grams of Cymodocea rotundata seagrass leaf extract, and then add magnesium powder, and then add HCl, and then homogenize. And then if the solution produce a red, yellow, or orange orange color, it is positive contain flavonoid. And then the last saponin test, a total of 0.05 grams of Cymodocea rotundata leaves extract, dissolve with distilled water, and then heat and shake, and then later up to 15 until 30 minutes. And then if the solution produce reserve form, it is positive contain saponin. And then the last method is antibacterial activity test. Tripsid soy agar media was poured into a petri dish and allowed to solidify. Aeromonas hydrophila and Vibrio harvey bacteria were inoculated on the media. This paper treat, and then this paper are on Tripsid soy agar media, and then sample were incubated at 28 degrees Celsius for 48 hours, and then the form inhibition zone is observed and measured by imaging. And then this is the formula for searching to inhibition zone, and then this is the result. Phytochemical screening test. In the following table, it can be seen tannin says the color is blackish green, and then the result test is strong, and then flavonoid test the color is yellow, and then the result is moderate, and then the last saponin test the result is warm until 15 minutes and then strong. And then this is the result of the t-test analysis on the inhibition of Aeromonas hydrophila and Vibrio harvey. In the following figure, it can be seen the largest of suppression zone is on positive control, and then the second is 10,000 part per million treat. And then this is inhibition zone for Aeromonas hydrophila and Vibrio harvey. In the following figure, it can be seen a black circle is 10,000 part per million treat, and then a red circle is positive control. And then conclusion. First, seagrass leaf extract of Cymodocea rotundata positively contain bioactive compound. And then second, the concentration of Cymodocea rotundata leaf extract and incubation time has an effect on the antibacterial activity of Aeromonas hydrophila and Vibrio harvey. The Cymodocea rotundata seagrass leaf extract can be used as antibacterial, uh, but uh, but weak for Aeromonas hydrophila and moderate for Vibrio harvey. Thank you for attention. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you, Miss Mila Santika, for your great presentation. We are going to the fifth presenter. We invite Mrs. Ilma Al-Adiani.
Yes.
Okay. Miss Mila Al-Adiani, you have 10 minutes for the presentation.
Okay, thank you for the invitation. Assalamualaikum warahmatullahi wabarakatuh. My name is Ilma Al-Adjani. I would like to present about my research with title Bioactive Compound of Seagrass Root Extract Thalassia hembrici as Natural Antibacterial Against Aeromonas hydrophila and Vibrio harvey Bacteria. Thalassia hembrici seagrass is one of the abundant seagrass in Indonesia with a density 34,089 individuals per 100 square meter. Thalassia hembrici seagrass contain bioactive compounds such as phenol, flavonoid, tannin, and saponin. The existence of seagrass Thalassia hembrici in Indonesia has not optimally utilized. Thalassia hembrici seagrass is one type of seagrass that can be used as antibacterial, antioxidant, antifungi, and antiviral. The use of seagrass as an antibacterial can be tested on bacteria Aeromonas hydrophila and Vibrio harvey bacteria. Aeromonas hydrophila bacteria is a bacteria that causes motile Aeromonas septicemia disease in freshwater culture fish, and Vibrio harvey bacteria, a bacteria that causes fibrosis disease in crustacea cultivation. In Indonesia, synthetic antibiotics are used to overcome disease problem, but the use of synthetic antibiotic is ineffective if used uncontrolled. Therefore, research on the bioactive compound Secretia Thalassia hembrici as an antibacterial need to be tested on fish pathogenic bacteria that was Aeromonas hydrophila and Vibrio harvey bacteria. The purpose of this research the first, to find out screening bioactive compound of Thalassia hembrici seagrass root extract, and to find out understanding the potential antibacterial of Thalassia hembrici root extract based on inhibition zone against two bacteria. Material and method. There are main material that I use in this research. That was water bath shaker, rotary evaporator, and incubator shaker. Research procedure that I use in this research. The first Thalassia hembrici root sampling, sample preparation, Thalassia hembrici root extraction with metamorpho analysis, and then the sample was screening phytochemical, and then rejuvenation and manufacture of bacterial suspend, and then testing for antibacterial activity based on the inhibition of zone, and then data analysis. Thalassia hembrici seagrass root extract, seagrass root conductly at Pangandaran Timur Beach. Pre-extraction activity, the first Thalassia hembrici sorting, and then the sample was washed using fresh water, and then separation of seagrass root from other part of seagrass, and then the sample was dried, and then was weighed, and then was dried and aired in an air-conditioned room, and then was weighed, and then was ground, and then next process extraction. Extraction activity. The first 40 grams of sample was put into the Erlenmeyer and then added to 200 milliliter of the methane for analysis was added into the air layer, and then the sample was homogenized with an incubator shaker, and then the filter was evaporated using a rotary evaporator, and then the filter was evaporated using a waterbed shaker. Phytochemical analysis. In this research, phytochemical compound tests are tannin, flavonoid, and saponin compound using screening method. Tannin test was done using as 0.1 gram of cigarette extract, and then added 2 milliliter of hot water, and then simmer for 5 minutes, and then filtered. And then the filter is added 10 milliliter FCl 1%, and then positive test of tannin marked the blackish green color. And then flavonoid test. Flavonoid test was done using as much as 2 milliliter of cigarette extract, and then added 2 milliliter of hot water, and then simmer for 5 minutes, and then filtered. And then the filter is added 0.05 milligram mg and 1 milliliter HCl, and then positive of flavonoid test mark the appearance yellow red until orange color. And then saponin test. Saponin test was done using as much as 0.05% of cigarette extract, and then heated, and then shake until foamy, and then there is foam up to 10 minutes. It means positive of saponin test. Rejuvenation and manufacture of suspend bacteria activity. The first, the test bacteria were take, and then were inoculate on media, and then incubation during 1.24 hours, and then the test bacteria were taken, and then were injected into a test tube, and then the bacteria was mixed, and then were put into the incubator shaker, and then the theoretic soluble media become cloudy. It means bacteria grow in media. Testing antibacterial based on the zone of inhibition. The first sterilization, and then synthetic soy agar media was poured into a petri dish and allowed to solidify. And then this bacteria were inoculate into the media with treat plate. And then this paper was treated according to next table. This paper was placed into synthetic soy agar media, and then sample were incubate. And then the prompt inhibition zone was observed and measured with images software. Antibacterial test treatment of Thalasseria cigarette extract against Aeromonas rubella and Harvey bacteria. Sample 1 is 10,000 ppm of Thalasseria root extract. Sample 2 is 5,000 ppm of Thalasseria root extract. Sample 3 is 2,500 ppm of Thalasseria root extract. Positive control is 10,000 ppm of ampicillin, and positive control is Aquades. Inhibition zone diameter can calculate with the following formula: is vertical diameter plus horizontal diameter per 2 minus well diameter. Inhibition zone diameter can categorize with low, middle, high, and very high. Data analysis. Data analysis can analyze with comparative description, Mann-Whitney test, and t-test. Result of this research of phytochemical screening, Thalasseria extract contain tannin, flavonoid, and saponin compound. Nothing indicate more convincing result according to Sibir et al. 2019. Result of antibacterial activity test of Thalasseria cigarette extract against Aeromonas and Harvey bacteria can use in figure A and figure B. Figure A is inhibition zone of Aeromonas bacteria, and figure B is inhibition zone of Harvey bacteria. The critical indicate the most influential treatment is positive control is ampicillin. Antibacterial activity test of Thalasseria cigarette extract against Aeromonas bacteria. The results show that ampicillin, the high inhibition zone in bacteria Aeromonas at good observation time 24 hours and 48 hours, compared to the treatment of extract group of 10,000 ppm, 5,000 ppm, 2,500 ppm, and negative control. Cigarette extract Thalasseria concentration of 10,000 ppm has the high inhibition zone in inhibiting bacteria Aeromonas at good observation time when compared to concentration 5,000 ppm and 2,500 ppm. Antibacterial activity test of Thalassemia secret root extract against Vibrio harvae bacteria. The results show that ampicillin had the high inhibition zone in bacteria Vibrio harvae at both observation time at 24 hours and 48 hours compared to the treatment of extract group of 10,000 ppm, 5,000 ppm, 2,500 ppm, and negative control. Secret extract Thalassemia concentration of 10,000 ppm has the high inhibition zone in inhibiting bacteria Vibrio harvae at both observation time 24 hours and 48 hours when compared to concentration of 5,000 ppm and 2,500 ppm. Antibacterial activity test of Thalassemia extract against aroma European and Vibrio harvae bacteria at the time of observation 24 hours and 48 hours show that all concentration sample extract have a relatively low antibacterial effect indicated by a range of the result. The conclusion of this research, the first root extract of Thalassemia contain tannin, flavonoid, and saponin compound, and the second, the antibacterial potential of Thalassemia root extract were categorized in low inhibition zone for both of bacteria. Thank you for your attention. Wassalamualaikum warahmatullahi wabarakatuh.
Thank you, Miss Ilma Al-Adiani, for your great presentation. We are going to the sixth presenter. Please, Miss Sahra Sapri Aulia. Miss Sahra?
Yes, sir.
Okay, you have 10 minutes for the presentation. The time is yours.
Okay, thank you, Mr. Moderator, for the time.
Yeah.
Kak Rama ini masukin PPT-nya gimana, Kak Rama? Nggak bisa.
Harus jadi host.
Udah host. I'm so sorry for this some error from.
Ini bisa.
Host, please present his presentation.
Yang mana cara ini ya?
Yes, please wait for a minute, Mr.
Okay.
Tuh, udah dulu.
Belum sih.
Udah dulu.
Okay, I'm so sorry for the error technique. First of all, bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Good morning, ladies and gentlemen. In this opportunity, I would like to, first of all, I would like to introduce myself. My name is Tiara Safira Aulia. I'm a bachelor student of Marine Science from Jenderal Sudirman University. In this opportunity, I would like to present about my research and team with the title Carbon Trap Estimation of Mangrove Vegetation using Remote Sensing in Segara Anakan Cilacap. Okay, ladies and gentlemen, beyond that global warming, now West Bank one of the more important issue in the world. We know that global warming impact have the impact all of the world. All of the country in the world get the impact of global warming. Beyond that, global warming can be happen because of the increasing of carbon dioxide in the air and following with the decreasing of the area of the forest in the world. Global warming cannot be avoided, but the impact of global warming can be minimized. How we can minimize the impact of global warming? We can minimize the impact of global warming by carbon trading. Carbon trading means that the forest country have to protect their forest, and So the forest can absorb the carbon dioxide in the air, and the industrial country have to pay for the forest country. According to Donato et al. 2011 said that mangrove forests can save up to 4 times more carbon than the other tropical forests around the world. Murdiaso et al. 2015 said that Indonesia is a country that has the largest mangrove forest in the world. So Indonesia can be play a role model for carbon trading. We know that mangrove forests can save up 4 times more carbon than the other tropical forests around the world. So we have to measure the potential of mangrove forests to absorb the carbon dioxide in the world. The measuring of mangrove forests potential to absorb the carbon in the field need a lot of time, cost, and effort. So we need some technology to minimize that and make it efficient. And the answer is by using remote sensing technology. Using remote sensing technology to determine the estimated carbon stock of mangrove forest, it will save time, cost, and effort. Okay, this is my purpose of my research. The first is to determine the extent and density of mangrove forest in Segar Anakan based on Sentinel-2A satellite image data. Second, to determine the potential of mangrove forest in Segar Anakan area as a carbon trap based on the area and density obtained from the Sentinel-2A satellite image data. Okay, this is my study of my area. The study is located in Segar Anakan Cilacap, and there I have 30 sampling points. And next is about the methods. In my research, I have 3 methods. The first is pre-survey. At pre-survey, I have to download the satellite image data, and next I have to correction radiometric, and next I have to crop the satellite image. The cropping of satellite image have a function to make sure if to focus at my research location, and next have masking. Masking is aims to spare the island and the water. And next I have the segregation of mangrove and non-mangrove. The segregation of mangrove and non-mangrove by using supervised classification with maximum likelihood classification. And next is calculation the area. The calculation of the area of mangrove is doing by software with EarthMap. And next, I have to calculate the density of the mangrove. The calculation of density with using NDVI algorithm. And next is survey. When I have done with the pre-methods, I have to survey in the field, and I have 3 tasks here. The first is identify the name of mangrove species and their number in plot transect. And next is measure the diameter of tree trunk at this height or dBH in a plot transect. And the last is take the percentage of canopy you cover using the camera from the below, like that. Okay, the last method is the method that was survey. When I have get the data from the field, I have to make an analysis. The first that I do is about calculate the leaf area index or LAI. The calculating of the leaf area index by using the image that I get from the field from the below of the tree. And next I have to calculate the biomass value. The calculate of biomass value by using allometric approximation. Allometric approximation, every species have a different allometric approximation. And next I have to calculate the value of carbon stock. And next calculate the absorption of value of carbon dioxide. And next I have to calculate the accuracy assessment. The calculation of accuracy assessment by using table confusion metric and root mean square error. And this is my allometric approximation. We know that every species have a different of allometric approximation with a different source too. Okay, ladies and gentlemen, next is about the result and discussion. The first is about mangrove area and density. Calculation of the area of the mangrove forest in Segar Anakan Cilacap in 2019 using Sentinel-2A satellite imagery obtained a total area of 5,481.6 64 hectare with accuracy 86.92%. The types of mangrove that found were Avicennia marina, Rhizopora mucronata, Rhizopora apiculata, Seriops tagal, Aegiceras corniculatum, Soneratia alba, and Brugueria gymnorhiza. And for the density, this is three class of density. The first is sparse density. Sparse density have with the green color with 706.92 hectare. And the next is medium density. The medium density have the yellow color with 2,526.8 hectare, and the high density with red color with the area of 2,848.16 hectare. Okay, the next discussion and result is about estimated carbon stock. Based on the calculation made, the total biomass value of mangrove forest in Segara Anakan Cilacap was 30,644.77 tons. The known value of mangrove biomass is used to calculate the value of carbon content in mangrove. Calculation of the value of carbon stock is done by multiplying the value of biomass by 0.46. So the value of mangrove forest carbon stock in Segara Anakan Cilacap is 40,096.16 ton or 2.57 tons per hectare. So we can make a conclusion that the mangrove forest in Segara Anakan Cilacap has the ability to absorb 9.44 tons per hectare of carbon. Okay, the last is about my conclusion. In this research, I have two conclusions. The first conclusion is mangrove forest in Segara Anakan Cilacap has an area of 505,481.64 hectares with the types of mangrove found, namely Avicennia marina, Rhizopora mucronata, Rhizopora apiculata, Seriops tagal, Aegiceras corniculatum, Soneratia alba, and Brugueria gymnorhiza. Mangrove with sparse density has an area of 706.92 hectare. At medium density has an area of 2,526.8 hectare. At high density has an area of 2,848.6 hectare. Mapping of mangrove density using NDVI index produces an accuracy of 86.92%. And the second conclusion is mangrove forest in Segara Anakan Cilacap have the potential to absorb 9.44 tons of carbon per hectare. And for the last, I would like to say thank you for Pusat Studi Bioscience Maritime LBPM Unsud for support my research. I think it's enough for me. Thank you for your attention. Assalamualaikum warahmatullahi wabarakatuh.
And the last presenter is Miss Titin Yulianti.
Yes, sir.
Okay, Miss Titin Yulianti, you have 10 minutes for the presentation. The time is yours.
Is my PPT has shown?
Okay, yeah.
Assalamualaikum warahmatullahi wabarakatuh. Thank you for having me in this seminar. Let me introduce myself. My name is Titin Yulianti from Marine Science Study Program, Jenderal Sudirman University. Today I will present our research entitled Characteristic of the Sea Level Anomaly during El Nino and La Nina in Southern Waters of Java. Climate change is a phenomenon of changing weather patterns that take place within a certain period. Climate change, one of the indicators of climate change is ENSO, or El Nino Southern Oscillation. Climate change affects on the frequency of ENSO event. This affects on the increase of ENSO. ENSO phenomena include El Nino and La Nina is formed as a result of sea surface anomalies in Pacific Ocean region. In equatorial Pacific Ocean region, one of the indicators of ENSO include
oscillation index, Oceanino index, and sea level anomaly. Sea level anomaly is the amount of deviation that occur in the average of sea level. The El Nino can reduce the sea level, meanwhile, La Nina can increase the sea level. Certain waters of Java, as we know, is a part of the Indian Ocean, which are influenced by many factors, such as monsoon, and so IOD, and South Java Current, South Equatorial Current, and Equatorial Countercurrent. Change in sea level during ENSO generally have a big impact on areas directly facing the coast. My research purposes are knowing the sea level anomaly during El Nino phenomenon in southern waters of Java, and knowing the sea level anomaly during the La Nina phenomenon in southern waters of Java. The method used in this research is the observation of sea level anomaly and sea surface current data from satellite altimetry. Data processing is done by extracted, sorted, visualized, descriptive analysis, and can be the result. This is my research site. Based on sea level anomaly analysis during El Nino in 2015, show that the sea level has decreased with an average of 0.075 meters. In June, El Nino is not seen yet. The El Nino began to appear from July to November 2015. The average decrease in sea level in July is 0.063 meters. In August is 0.070 meters. In September is 0.180 meters, and this is the highest decrease in sea level. And in October is 0.142 meters. And the last November is 0.112 meters. The decrease in sea level occurred due to the weakening of trade wind and a shift in the warm flow from Indonesia to the east of Pacific Ocean. This is also due to the void of water masses around the water surface as a result of displacement of water masses to each side of West Indian Ocean and East Pacific Ocean. This condition also causes the upwelling intensity to increase. Based on sea surface current pattern during El Nino, it can be seen that during El Nino, South Equatorial Current was strengthening. South Equatorial Current pushed the water masses westward from southern waters of Java, and this is causing decrease in sea level. Next, in sea level anomaly analyzed during La Nina in 2016, show that the sea level has increased with an average of 0.298 meters. And in June, the average of increase is 0.422 meters, and this is the highest increase in sea level. And in July is 0.306 meters. In August is 0.221 meters. In September is 0.254 meters. In October is 0.310 meters. And the last in November is 0.267 meters. The increase in sea level occurred due to the trade wind was strengthening. Also, the warm flow was shifting from the East Pacific Ocean to Indonesian waters, especially southern waters of Java. The increase in sea level during La Nina cannot be separated from the accumulation of water masses that moving from the West Indian Ocean and East Pacific Ocean to Indonesian waters. This condition also caused downwelling intensity to increase. And this is the surface current pattern during La Nina 2016. We can see that equatorial countercurrent was strengthening. And this current has caused the accumulation of water masses because it is pushed by water masses from the eastern part of Indian Ocean and causing an increase in sea level. And this is the table of monthly sea level anomaly, include minimum, maximum, and average of sea level anomaly. And next, based on fluctuation of South Oscillation Index El Nino 3.4 index during ENSO, it can be seen that from June 2015 there was El Nino, and from June to November 2016 there was La Nina. And the conclusion from this research is during El Nino phenomenon in 2016, the sea level of southern waters of Java has decreased, and anomaly value is negative with an average 0.075 meters. The south equatorial current was strongly and pushed water masses westward, caused a decrease in sea level. And during La Nina phenomenon in 2016, the sea level of southern waters of Java has increased. Our anomaly value is positive with an average 0.298 meters. The equatorial countercurrent was strongly caused a build-up of water masses in southern waters of Java. And this is for me. Thank you.
Okay. Thank you, Miss Titin Yulianti, for your presentation. And now we will have a discussion section. The audience may ask questions by activating the raise hand or writing question in the chat column. Okay. There is question from Shirley Puspita Ningrum or Titin Yulianti. What is the impact of La Nina for Indonesia?
Okay, may I answer the question? The impact of La Nina for Indonesia. The first is the increase in sea level, as my present before, and second is the La Nina can cause the heavy rainfall. And BMKG analyzed based on historical record that the La Nina can cause an increase in accumulated amount of monthly rainfall in Indonesia, and it is to 40% above normal. And the normal is to 2,000 until 3,000 millimeter, and because the La Nina, it can increase until to 40% above normal.
Okay. Thank you, Ms. Titin.
Thank you.
Is there any question? Oke, audien mungkin bisa bertanya dengan menggunakan bahasa Indonesia. Apakah ada yang ingin bertanya? Hello, any question? Ada yang ingin bertanya? If there are no question, I want to ask Miss Sahra Aulia. Miss Sahra,
Yes, sir.
I want to ask for you, is there any certain characteristic required to determine your 30 sampling point?
I'm so sorry for the characteristic of mangrove or the other parameters.
Yes.
I'm so sorry, Mr. Moderator. Can you explain what the kind of the parameter that you asked for that?
It was said that mangrove area in Sagaranakan can absorb 7.4 ton of carbon per hectare.
Yes.
With that potential, is there any treat that could reduce the mangrove area in Sagaranakan?
Okay. Wait. So, Mr. Moderator, means that what is the location of the have the bigger absorption?
Hello,
Hello.
Mrs. Jahara?
Yes. I'm sorry, Mr. Can you repeat your question? It's not clear the sounds.
Oh, okay. It was said that mangrove area in Sagaranakan can absorb of carbon per hectare. With that potential, is there any trait that could reduce of mangrove area in Sagaranakan?
Yes, mangrove in Sagaranakan can absorb
9,44 ton.
Yes, 9.44 tons carbon per hectare.
Apakah ada ancaman dapat mengurangi area mangrove di Sagaranakan?
Oh, okay. I understand for your question, Mister. For the untuk ancamannya itu, boleh saya bilingual?
Oke.
Baik. Mungkin untuk ancamannya itu pengurangan luasan lahan dari mangrove-nya itu atau dari itu diakibatkan oleh beberapa hal, diantaranya yaitu penebangan hutan yang kita ketahui seperti illegal logging. Ada juga penelitian dari Purwanti et al. 2014 kalau nggak salah itu menyatakan bahwa luasan hutan mangrove di Sagaranakan pada setiap tahunnya itu mengalami penurunan secara terus-menerus. Ada juga yang diakibatkan oleh karena adanya tingginya sedimentasi yang menyebabkan si hutan mangrove ini terdegradasi. Seperti itu. Jadi ada ancaman yang dapat mengakibatkan penurunan dari potensi si mangrove untuk menyerap karbon. Itu diakibatkan karena adanya penurunan dari luasan hutan mangrove tersebut.
Oke, thank you. Sarah.
Okay, thank you, Miss.
Question from Viviani to in your research on methanol.
Okay. Thank you for question. I will answer a bit for your attention. Why I use methanol pro analysis in this research? Because methanol is an effective solvent to check polar and non-polar bioactive compound.
You.
Okay. I want to answer Miss Viviani question. Why I choose methanol solvent in my research? Because methanol is a universal solvent, so as to be able to dissolve its polar or non-polar analyte. Methanol can attract alkaloid, flavonoid, and sulfoname from the plant. Thank you.
Okay, thank you. There is question from why did the highest decrease in sea level occur in September during El Nino?
Okay, thank you for Ms. Agnes for the question. Why the highest decrease in sea level in September during El Nino? It because influenced by southeast monsoon. When southeast monsoon wind blow in July to September, the South Equatorial Current, which from in the Eastern Indian Ocean, will flow westward. Then its position will shift beyond latitude 10 degrees south, and it will reach the southern coast of Java Island. And the existence of this monsoon can cause the speed of South Equatorial Current to be accelerated, and this is why the highest decrease in sea level occurred in September.
Okay, thank you. Apakah ada yang ingin bertanya lagi?
Mr. Moderator, may I ask to Titin?
Mr. Tenggono.
Okay, thank you. Titin, could you hear me?
Yes, I hear you, Mr.
Okay, Titin, in your presentation, if you have mentioned if the INSU is the phenomenon which happen in Pacific Ocean. And my question is how the INSU mechanism can affect sea level anomalies while your research allocation is in the Indian Ocean. Thank you, Titin.
Thank you for Mr. Mukti Tronggono for the question. How mechanism of the ENSO can reach the southern waters of Java? It's because from Indonesian troop flow.
Is it only ITF, not the other mechanism?
And the other mechanism is from the...
I'm sorry, Mr. Mrs. Titin. Please turn your microphone and video. Your video.
White in my room, the lightning is bad.
Okay. Thank you, Miss Titin.
Beside of Indonesian strip flow, there are air pressure that move from Pacific Ocean region to Indonesian region that carried the water vapor, and when the La Nina, it can be cause the heavy rainfall.
Okay, Titin. Yeah. That's answering. Thank you, Mr. Moderator.
Okay, thank you, Mr. Mukti Pranggana.
Thank you.
Any question?
Ini tahu kan?
Okay, finally, we have come to the end part of your discussion. We would like to say thank you again to the speaker for the informative and interesting talk, and the audience for active participation. Hopefully, the presentation will be beneficial for everybody. For the closing of this seminar, I would like to excuse myself and give the time back to Mrs. Diarudi as the master of ceremony. Warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh.
Yes. Thank you very much, Mr. Amfitriadi MP, for getting the very interesting session of the parallel session. So we've talked about the bioactive compound. We also talk about the what is it, the carbon that's inside a mangrove ecosystem, and etc. Well, ladies and gentlemen, so before we go into the expert speaker session, unfortunately, because of the time difference between the invited speaker time uh, with Indonesian time, therefore, we might have a few minutes gap of time. Therefore, we would like have a time break. But before that, maybe it would be better if we have a photo session for the parallel participants. So for everyone here in a parallel session, could you possibly turn on your camera? Yes. Ya, silakan hidupkan kameranya, turn on your camera, and give your best smile. All right, are you ready? The committed, please take the picture of the participant. So, okay, I saw here some of the participants still turn off the camera. Could you please turn it on? Uh, there's Miss Maria Menita, Etlin Zakia, and etc. All right. 1, 2, 3. So we have two slides. Yes. Give you a best smile. 1, 2, 3. Okay. Thank you very much. And I have the information that Professor Slamet Priyatno already in our Zoom room. Assalamualaikum. Good afternoon, Professor Slamet.
Yeah, good afternoon. Yes, I'm with you now.
Thank you for joining S2FiMaS. And
Okay.
yes. So as we know, because our first expert speakers are already in the room, so we are going to start the session, or we will have a break. All right. Yes.
You, Atu actually. I can session now or later. It's,
All right. Would it be all right if we start the session now, Professor Slamet? Hello?
Yep. Okay.
Yes. Okay. I will introduce the moderator first before you share.
Okay.
Distinguished guests, ladies and gentlemen, I believe that all of you are the seminar can't wait for this upcoming session. So because we will have 3 expert speakers that will provide or deliver their talk today. They are Professor Slamet Budi Prayitno from UNDI, Iri Patokta PhD from Czech Republic, and Dr. Marike Hans from Bochum, Germany. So as usual, this session will be led by moderator. But before the moderator holding the session, allow me to read his curriculum today. He is a lecturer in an aquaculture department of Fisheries and Marine Science Faculty, Jenderal Sudirman University, and he obtained his doctoral degree in Germany. His expertise is in fish disease and immunology. Let us give him a warm welcome, Dr. Rernat Hamdan Shakuri. So to Dr. Hamdan, the time is
Yeah. Thank you very much, Ibu Ruri. Is my voice clear enough from your side?
Yes, it is clear.
Okay, thank you very much. The Honorable Dean and Vice Dean of Fisheries and Marine Science Faculty, and the Honorable the Chief of the Committee and also the member of this seminar series, and also the Honorable Professor Budi Prayitno as our expert speakers. The Honorable ladies and gentlemen, good afternoon. Thank you very much for the Master of Ceremony for introducing me. I am the moderator of this session, and for the first discussion, we have here Professor Slamet Budi Prayitno. Please, the committee make sure that our expert speaker is arranged as co-host. So It is the CV of Professor Slamet Budi Prayitno. He is a professor in University of Diponegoro, and his expertise is in fish diseases, as far as I know, and of course, he has great experience in fish diseases, and I agree that one of the people in Indonesia who could summarize current disease in fish aquaculture is Professor Budi. And he also experts of Indonesian government in fisheries development, especially in aquaculture. But also he has unique activities. I'm sorry to say that. Yeah, it is an interesting because he has activity in Boy Scouts or Pramuka, right, Professor Budi?
Yes, yeah.
Yes. And also another interesting, he is Ketua Badan Penyelenggara Senam Sehat Indonesia Jawa Tengah.
Yeah.
Yes, it is very interesting for us. And for this seminar series, Professor Budi would like to talk about antibiotic in aquaculture, especially in Indonesia. So antibiotic could be parasitic or commensal organism living on the surface of another organism or inside other organism. And as we know that the parasitic one could cause ecological losses in fish or in fish organism, and also economical losses, especially in aquaculture. And for this session, Professor Budi have around 30 minutes for the talks, and then we have around 10 minutes for the discussion. And as moderator, I would like to strongly suggest for the participants, please during the talks, you could type the question in the chat room of this Zoom meeting, and also in the chat of the YouTube. I think I could manage to access both of the chat rooms. So, Professor Budi, please welcome.
Thank you very much, Pak Hamdan Guri, for your very nice and very good introductions. Actually, today my topic is not really related to disease, but more to the commensalis and antibiotics as food for culture fish. My presentation in Bahasa Indonesia because I knows that most of the participants is from Indonesia. So just to make everybody easy to understand.
Yes,
First, the introduction. We all know that the intensifications of the aquaculture have already increased the production and productivity very significantly. And in 1916, for example, in 2016, Indonesia is able to produce 4.95 million tons of fish from the aquaculture. And in the next 10 years, in 2030, we expected to produce more than 10 million tons. So the concerns of these intensifications and production target up to 2030, first, most of people now try to move their activities, especially for the brackish water aquaculture development from coastal area to further inland area. It means that there is a risk of the waste of the seawater into the land area. We can see actually this case in the southern part of Java, for example in Purworejo, Kebumen, which is paddy field, and also banana plants start to die due to the waste of the shrimp ponds activities. The second one is about the pollutions carried by the shrimp intensive activities in many areas. And the third one, the usage of fresh water, especially when the activities move from the coastal area into inland area, then the usage of fresh water become very high, especially where they are using borehole waters. And of course, the cost is relatively high. Although if we talk about shrimp business, if you are succeed in the first crop, you can still fail 1 or 2 times without any problems, because the benefit or the profit almost 3 times of your operational cost. So this is the reasons why I really choose these topics, because it is quite interesting. And then of course there is some approach that has been done. First is about how to minimize water exchange, and how to use the microorganism to absorb the nutrients waste extracted by the animal and unused feed by pre-colonizations of microorganisms and the applications of biofloc and biofilm. My presentation today actually concentrated on the biofilm, although the title is about epibiosis or epibiotic. Epibiosis was a word from the Greek. Epi means surfaces and bios is life. So the epibiosis is organism or microorganism from any individuals that live in the surface, in the substrate, and they can be basibion and also epibion. So epibiotic actually is what you call it, a groups or a consortiums of many microorganisms that it's non-specific and can be also sessile organisms that may live together as a basibion or epibion, and they are attached and then grow on the surface of the substrate, and when they grow up, they could be a biofouling and also biofilm. Biofilms is an association or consortium of the microbial organisms which have a substance of polymeric extra shell that responsible in the encycles and also geochemical cycles. It means that biofilm is surely the consortium of microorganisms that absorbs or used the anorganic materials from the nitrogen. How the biofouling and biofilms occurs. Both micro or macroorganisms, macroorganism possibly periphyton, yeah, the organism that they on their life cycles, they need substrate to attach, yeah, and they usually attach on the dead surfaces. Can also be in the live organisms, whether this is a plant or possibly animals, and they become a biofoulings. It's called biofoulings because they really destruct the activities, the growth, and possibly the metabolisms of the host. And the second one is biofilm. Biofilm actually a very thin layer in the surface of the substances or substrate where organism was live, and we usually could feel it if we have some substrate or particles or possibly whatever the everything in the water for more than 7 days, then we feel very smooth and a bit slippery. This is the production of the microorganisms. In the seawater, there are at least 10 to 100 larvae of microscopic organisms with around 1,000 fungi, 1 million bacteria, and over 10 million virus. It means that the pressure of the environment to this organism is quite high, and we could imagine how the aquatic organism have to neutralize this all of foreign organism or foreign body to be neutralized in order to prevent any imbalances, any disease, or any infection. This is the very interesting thing about the aquatic animals, because the aquatic animal has possibly 100 to 1,800 times difficult compared to the terrestrial animal in terms of how they hold to be survive, growth, and follows their life cycles. When this microorganism was attached to the substrate, then they will colonize and then compete each other, dominate the area, then they will form biofilm. This is an example of one of the students from the Hasanuddin University on his bachelor scripts. He found that at least in terms of macroorganisms, at least there are 7 to 8 classes of the organism attached to the we called substrate or dead substrate made from wood, yeah, in the dermaga. This is actually a demonstration of how organisms are really rely on the surface of the substrate, whatever the substrate, in order to get food, to attach, to develop, and to complete their life cycles. So when we talked about biofilm, actually biofilm involves the competition among the microorganisms who try to attach, to live to replicate in the surface of the substrate. And in the past research, the predominant species was found from the genus Pseudo-Alteromonas. Why? Because these genus are able to excrete antibacteria in order to colonize the host. The antibacteria excreted from this genus is agarices, toxin, bacteriolytic, and several enzymes, where the microorganisms itself, with this antibacterial, with this enzyme, with these toxins, were the way to prevent from the predators and also to protect themselves from the grazing. It means that the higher organisms will eat them. But if we talk about the abiotic biofilms, actually this is the organism that quite easy to find, as I explained before, and they also have a role in the water, in the cultural media, which we could use it as a beneficial rather than a threat. I think Pak Hamdan Shakuri have already explained that abiotic can be parasitics, can be commensals, yeah? And in my view, the parasitics, abiotics is an opportunistic bacteria mostly. It means that they become a pathogen where the environment is given the way to be pathogen, especially in the stressful environment or when the hosts are in the stress conditions. And water is a food factor for the sessile organisms which attach to the surface of the substrate. And the organisms which become biofilms like bacteria, protozoa, diatoms, actually is a source of a food for the past. Polychaete, macroalgae, echinodermata, crustacean, sponge, and tunicates. This is also the place where the meroplankton was attached in order to complete their life cycle to be a juvenile. So when we culture the water or culture media, we could use this as a group of organisms that will use the metabolism waste or other wasted produced by the cultural fish. So I really like to see these abiotics as a source of food with high protein for the cultural organisms, especially fish. Because these bacteria, these microorganisms, when culture fish was eat this kind of food, it will increase their immune system. And on the other hand, water quality could be hold as good as, or could be hold in a very good conditions. Although we aware that in these biofilms, bacterial opportunistic bacteria was present there. And therefore, if we follow the recent technology in the intensive shrimp culture, biofilms is one of the substrate of the organism which mostly avoided by the shrimp culture. However, if we concerns about non-intensive aquaculture, if we concerns about traditional aquaculture, biofilms is one of the way to reduce food conversion ratio, to reduce cost of feed, because we can use this as a source of natural feed. So biofilm could be very beneficial for the aquaculture if we could really select the culture species which have the behavior of grazing. Otherwise, the benefits is lower. For example, if we compare fish and crustaceans, the biofilm is more beneficial to fish rather than to crustaceans. This is because they have different way of they different have a feeding behavior. Udang-udangan or crustaceans, they are chewing, while fish can grazing, especially for the omnivora or fish like carps and niloticus who are grazing on the surface of the ponds. So why I said this is a very good natural food, natural feed, because the protein contents is between 23 to 30%, and it's contain microalgae and heterotrophic bacteria, yeah, which I have said before. It's huge of the immune, what you call it, substances that could promote the immune system of the host, and can be also as a growth promoter because they contain the bioactive substances that could also enhance the growth and the fertility. Also, we usually increase the resistance from to disease. Some research, I will just show you one research done by a PhD student in Europe. Yeah, that they have tried, the Indian, the people from Europe, they have tried to demonstrate the benefit of biofilm in the aquaculture, especially for the freshwater and also for the brackish water aquaculture. And biofilm has proven increased growth, food conversion ratio, survival rate, and also improve the water quality and reduce the organisms, falling organisms. That has been tried in the Icanila or Niloticus, tilapias, carps, and Labiorohita, and also we call it the orange fin labio, and Penos monodon, Lithopenos vaname, Macrobacterium rosenbergii, and also from the Merguensis. It means that in the traditional and semi-intensive aquaculture have been tried in freshwater and brackish water activities. This is one of the PhD research from the Audiello et al. 2011. That in terms of the biofilm, they try to compare artificial substrate and without artificial substrates for the shrimp Penaus vaname that he tried in the traditional plus aquaculture. It is demonstrated that the growth and the final growth or final weight of the shrimp was much better with the artificial substrate compared to without artificial substrate. Even in the higher stocking density, this artificial substrate still demonstrated, or biofilm in the artificial substrates demonstrated better growth and final weight compared to the without artificial substrate. The food conversion ratio is also better compared to the non-artificial substrate and both stocking density. When we look at in little bit detail, we show that the water quality parameter, especially total ammonia nitrogen, with a stocking density 600 and 800 with artificial substrate compared to without lower. It means that biofilms, microorganisms, the biofilms, is really involved in the reduction or in the usage of the nitrogens released by the culture fish or animal cultures. This is also shown for the unionized ammonia and also nitrate. This is indicate that when the nitrate was higher, mean the toxic ammonia was utilized by the microorganisms of the biofilms to convert from the toxic into non-toxics, and finally become the nitrate is a food for the primary producer. From these demonstrations, it seems that with the biofilms in the traditional and semi-intensive aquaculture could reduce the dangers of the toxic substances released by the culture species. And we do hope that the mortality due to the water quality could be reduced with the biofilms, and we also provide good quality of feed for the culture animals. The survival rate also higher. Food conversion economics also much better. So that's why I talk this, because in the traditional and semi-intensive, they are still rely fully on the natural feed or artificial feed. Not many farmers using biomes as a source of food. They thought rather than they thought the biofilm is a danger rather than a source of food. So I actually ask the audience to try to explore this opportunity or these resources in order to reduce the cost for culture. This is the identifications of the second layer. The first layer is producer, bacteria, and so on. But the second layer of the biofilms who live in the biofilms is diatom, ciliate, flagellate, nematode, copepod, rotifer, which this is all is natural food, a very delicious food for fish, and therefore biofilms, yeah, actually biofilms, yeah, actually is a very good food. Although again, we have to really maintain the water quality in a very good condition in order to prevent the opportunistic bacteria not to convert into the pathogenic bacteria. My last
Okay. We have
presentations
five minutes.
is actually there's two things just to remind us the biofilm is not really applicable for the intensive farm, especially in the shrimp farm, because abiotic biofilms is rather a danger rather than a food for shrimp. Therefore, if we look at to the intensive shrimp culture, they are using bioflocs and probiotics as a source of food and a source and to maintain the water quality rather than biofilm. This is because shrimp is not really grazing. And the second one, uh, in the intensive culture, especially for the shrimp intensive culture, waste from the unfeeding food, and secondly from the what you call it molting is quite high. So biofilms is not appropriate way to use it in the intensive culture. Therefore, bioflocs and probiotic are more used. But in the fish intensive culture, on the other hand, antibiotics biofilm can still be used because we, although in a very intensive culture, we still give space for the fish culture or culture fish to behave naturally. They have the behavior of grazing. So we give the space for this behavior. And in line with this, we also provide very good quality of feed in order to improve their immune system. Of course, the impact will be grow faster, health status better, food conversion better, and also better survival rate. Okay. As a conclusion, first, abibiotics is a microorganisms which live in the surface of the substrate. They form the community interspecies. Abibiotic can be a biofilm or biofloc or also biofilm, which is biofilm can be formed when higher animal was live there, and also phyton was live there to consume microorganisms in the surface of the substrate. Biofilms is a consortium of microorganisms autotrophic and heterotrophic, which is form the complex nets where the unorganic nitrogen excreted from the cultured animal will be assimilated by microalgae, autotrophic bacteria in the surface of the substrate, and then consumes by the detritus and other primary consumer. Abibiotic biofilm is a source of food. It's very good source of food And the last one, biofilm, is a good to be applied because it is very cheap, easy, and applicable, usable for the farmers that they don't have modality, capital to improve that scales of the aquaculture from traditional into semi-intensive or from semi-intensive into intensive aquaculture. I think this is all that I can share on this seminar. Thank you very much, and I return this time to moderator. Please, Pak Hamdani.
Yeah, thank you very much, Prof. Budi. I'm really sorry that I didn't really expect that you will be talking more about the commensal antibiotics.
Yeah.
But it is really interesting, and I think we will see in the future, and we hope, of course, that this strategy will be applied in our fish aquaculture, because as we know that now the people, especially in shrimp, more likes to do the intensive one. And
yeah,
yes, of course, there is a big problem with the waste, of course. Thank you very much for the presentation. And now we have 10 minutes for the discussion. So please, participants who would like to ask questions to Professor Budi, first you could type your question, and also you could ask your question directly in this forum. So please raise hand.
Okay. If there is no question, then thank you very much.
No,
Hamdani, yes, okay, no problem.
I will have.
I can ask in the Bahasa Indonesia, it's okay.
Yes, yes. So for the student, you could ask the question in Bahasa. If not, I have at least one question, Professor Budi. So as we know that the formation or the development of the biofilm is occur in cycles. I mean, there is cycle of colonization and biofilm, and then will come the death phase. So what is the strategy to deal with this cycle of biofilm development in aquaculture farm?
Yeah, okay. Thank you very much, Pak Hamdani. Yeah, actually, there is two approach. Yeah, one is we naturally culture the biofilm without any injections of the species or preferable species. But the second approach was we could induce preferable microorganisms into the culture water in order to develop the biofilm. Why? Just to ensure that the biofilm which grow and colonized the surface of the substrate is a microorganisms that predominantly microorganisms is a what we call it good microorganisms. Yeah. So the biofilm is really could be controlled as a source of the food. I think this is the approach. That's why in some aquaculture business, Hamdani, especially for the ornamental fish with low stocking density but high value, they already use the biofilm as a source of the food and a way to improve. Some microorganism species, they usually gram-positive bacteria, so like bacillus, yeah, and also some pseudoalteromonas, which is selected non-pathogenic one, and with the hope that they will colonize and dominated the surface of the artificial substrate. This is that has been done, Pak Hamdani. Yeah.
Okay. Thank you very much, Professor
Thank you.
Budi.
Yep.
So once again, for the audience who would like to ask question, you could unmute your microphone and ask your question directly to Professor Budi. Yeah, hello.
I have one question.
Okay,
I'm back.
please, Bu Ruri.
Yes. Good afternoon, Professor Budi. So, as you mentioned that the epibiotic also can contribute as an antibacterial. So, if we would like to isolate the antibacterial compound from the epibiotic, what is it, biota? Should we do the, what is it, the common extraction method, or there is a special things to do before we do the extraction? Thank you very much.
Okay, thank you very much, Ibu Jaruri. Yeah. The first one, how we isolate the microorganisms from the biofilm. Actually, we talk about the methodology is the same with the methodology that we use. Depend on what kind of bacteria that we'd like to isolate it. Because in the biofilm, there is in general, there's two bacteria, gram-positive and gram-negative bacteria, that they need the certain media. And also in the biofilm, there is Nitrosomonas and Nitrobacter bacteria, or we call it nitrification bacteria. And in some cases, it's depend on the depth of the ponds. They usually also contain a facultative bacteria, means that they also in an anaerobic or aerobic bacteria. Therefore, the media is really depend on what kind of the bacteria that like to isolate it. The method, they usually using the general bacteria that they create to collect it, or TCBS for the marine and this water. But again, with this common media, they usually the gram-negative bacteria grow much faster, and the gram-positive bacteria very slow growth. So we usually miss this slow growth bacteria, which is most of them are really what we call it good bacteria for the extraction of absorption of the substances from the nitrogen. So there is no certain media, Ibu Jahruri, yeah. But in my experience with the friends who work with the what you call anaerobic bacteria, we should use really certain certain media, which is not really isolated in the, we call it in fume covered with the oxidation with the air, but the water or the substrate was scrapped inside the water and directly put in the what you call it, flasks, and then tutup, and then just leave it for a week, and then there is a tube then put it in water tube in order to produced by this bacteria. This is for anaerobic. And we then found that this anaerobic bacteria actually is quite massive in numbers, and they very high contribution in productions of ammonia. Therefore, if we'd like to know so many ammonia is really produced by the microorganisms based on the deteriorations of the uneaten food and so on, then we have to use this method. Then we can compare this volume of ammonia produced with the number and the populations of bacteria who used and converted this ammonia into nitrate and nitrate. This is one PhD work with this one. Actually, at the time, he works for oil degradation bacteria and also the ratio between the anaerobic bacteria and aerobic bacteria in order to know the speed of the absorption or the use of the ammonia produced by this anaerobic bacteria. So I think this is my answer, Bu Juari. It depends on what are you going to see. But if just collecting, we could use the common media. But please subcultivate 3 or 4 times in order to give the slow growth bacteria to show up. Yeah, they use a gram-positive bacteria like Streptococcus, for example. They grow for 5 to 7 days, yeah, and a very thin layer, very thin and bening, yeah. So usually this kind of colony was missed when we just concentrated on 24 or 48 hours bacteria which grow in the media. Mudah-mudahan puas.
Ya, terima kasih Bapak.
Thank you.
Okay, thank you very much Bu Dia Ruhuri and Pak Slamet Budi Prayitno. Actually, it is very interesting discussion because for me it is a new strategy how to use epibiotics to increase productivity of aquaculture, especially by reducing the cost of artificial or commercial feed, and also increasing the immune system of the cultivan organism. And I think it is really a good strategy in fish aquaculture. Unfortunately, we have to finish our discussion, Professor Budi, because we have time limitation. Once again, thank you very much for your talk and also for the nice discussion.
Thank you, Pak Hamdan.
Please, ladies and gentlemen, our applause for Professor Budi.
Thank you. Thank you for all audience. Thank you, Pak Hamdan, as a moderator. See you.
See you. See you.
Bye-bye.
Bye.
Nggak foto nih.
Ono gajah barang nang kono. Sorry.
Siap.
Langsung bahasa Banyumasan kali ya.
Mas Bud, foto lah Mas Bud.
Oke, siap.
Mau foto dulu ya. Monggo panitia foto.
Nah, kangen sama Mas Budi, sudah lama nggak
ketemu. Siap, iya lama nggak ketemu ya. Aduh.
Ini di Semarang atau di Pandak, Prof?
Di Semarang. Tahun baru nanti ke Pandak.
Mas Bud, nanti persiapan saya dibantu ya Mas Bud.
Siap, oke, pasti. Don't worry, selalu. Oi, Amron, monggo.
Sudah, panitia?
Oke, panitia sudah.
Panitia, monggo dijepret. Mas Budi senyum lah.
Oh iya, pastilah, senyum terus lah. Nggak dikira repair rekasan, aku rasa senyum.
Baik, terima kasih.
Terima kasih.
Terima kasih Pak Budi, terima kasih.
Sama-sama, terima kasih semuanya.
Ya.
Yuk.
Ini kita masih menunggu.
Ini gimana? Masih menunggu?
Ya, saya baru kirim ini email tapi belum dibalas. Mungkin langsung ini kayaknya join. Nanti kita coba lihat.
Yang penting cadangan siap terus. Stay terus.
Kalau nggak nanti terpaksa Pak Hari yang presentasi gitu kalau nggak ada.
Kalau begitu saya sholat dulu ya.
Oh ya silakan Pak Amda. Nanti kita artinya fleksibel aja. Nanti kalau udah masuk kita sapa dulu nanti.
Bu Ruri, saya sholat dulu ya Bu Ruri.
Oh ya baik, silakan Bapak.
Mas Budi manggilnya gajah.
Ladies and gentlemen, so we have approximately 10 minutes break while waiting the next expert speaker to join the Zoom room.
Butuh sholat ngomong siap.
Dosen awak ngomongin di berani tinggal.
Kemarin tim apa namanya? Tim korespondensi waktu ngirim surat ke Jiri dikasih nggak apa namanya? Link Zoom-nya gitu. Sama mereka.
Right, while waiting the organizer to find some video profile. We would like to appreciate your attention and also your participation.
Suaranya.
To the seminar. Thank you for joining the S2FiMaS seminar series. So this is the 4th series. We hope you have a pleasant seminar and enjoy the seminar until the end of this event.
Suaranya turun naik.
Sebagai salah satu universitas yang terletak di Kabupaten Banyumas, Universitas Jenderal Sudirman turut memiliki peranan penting dalam perkembangan dan pembangunan pedesaan di Kabupaten Banyumas. Fakultas Perikanan dan Ilmu Kelautan yang menjadi salah satu bagian dari 12 fakultas di Universitas Jenderal Sudirman juga turut bersinergi menerapkan tridharma perguruan tinggi, yakni pendidikan, penelitian, dan pengabdian masyarakat pada sektor perikanan dan kelautan di Kabupaten Banyumas.
Fakultas kami mempunyai visi untuk menjadi fakultas yang unggulan dalam bidang ilmu perikanan dan ilmu kelautan berbasis kebutuhan masyarakat yang dalam rangka mengelola sumber daya hayati secara lestari dan menurut kearifan lokal. Untuk mewujudkan visi ini kami menyelenggarakan beberapa misi, yaitu misi yang pertama adalah menyelenggarakan pendidikan yang berkualitas atau berkualifikasi unggul untuk seluruh program studi yang ada di fakultas ini. Kemudian juga untuk meningkatkan pengelolaan fakultas yang lebih baik, dan beberapa langkah yang sudah kami lakukan antara lain adalah pertama, penelitian di bidang pemanfaatan sumber daya laut, yaitu rumput laut yang diteliti ada beberapa dosen kami yang bertujuan untuk menghasilkan bahan bakar minyak yang berbasis sumber daya kelautan. Dalam hal ini adalah rumput laut. Kerjasama internasional maupun lokal dengan institusi lain di dalam maupun di luar negeri itu sudah kami lakukan juga dengan beberapa universitas di Korea Selatan, di Jepang, kemudian di Prancis, dan beberapa negara lain. Kemudian pelaksanaan kerjasama ini dalam bentuk misalnya penelitian bersama atau juga pertukaran dosen. Kami berharap tentunya pada seluruh calon mahasiswa untuk bergabung ke
Thank you. Okay. Once again, hello, Dr. Jiri Patoka.
Hello, nice to meet you.
Nice to meet you. My name is Hamdan Sakuri, and I am the moderator of this session. So, first of all, let me introduce you to our audience. So, please, the committee, please display the curriculum vitae from Dr. Jiri Patoka. If not ready, then I would like to introduce you based on your CV. So, ladies and gentlemen, in this afternoon, we would like have the next talk and discussion with the second expert speaker. His name is Jiri Patoka. He comes from Czech Republic, and he is PhD in General Animal Science, Czech University of Life Sciences in Prague, Faculty of Agrobiology, Food and Natural Resources. Currently, he is an associate professor in the same university, especially in the Department of Zoology and Fisheries. He has experience in research activity in Indonesia, especially in the project of Alphabet. I do not really know what is Alphabet, but it is from Erasmus Mundus Action 2, and he is he was a principal investigator in Bogor Agricultural University. He studied mostly about the crayfish. So in this time, he would like to present about the current status of crayfish in Indonesia. And another thing about his curriculum vitae, I saw in the YouTube that he was a lecturer in Universitas Terbuka. Is this right? Dr. Chiri? Yeah, you are a lecturer in Universitas Terbuka, and I think it is also one of them about the crayfish. So for this seminar, there will be 30 minutes for your presentation, and then it will followed by 10 minutes of discussion. So Dr. Jiri, time is yours. Could you present, do you have ability to present your PPT?
Yeah, I will share my screen.
Please, Kamite, help him and arrange. Ah, yes, it is. It's okay.
Is it working?
Okay. Yes, it is working.
Yeah, perfect. So, hello everyone. Thank you very much for inviting me, giving me the opportunity to present here our outputs about crayfish in Indonesia. The research is based on cooperation with the Bogor Agricultural University, IPB, and also other universities like Terbuka and General Sudirman University. Especially, I'm in cooperation with Romanus Edi Prabowo. So, hi, Romy. Now, about the current status of crayfish in Indonesia. Here is a map of Indonesian territory. As you know, there are 3 biodiversity hotspots. It's important because the biodiversity richness is similar to this is one of the most important regions globally. Sundaland, Wallasa, and Sahul. Crayfish, freshwater crayfish, in Bahasa Lobster air tawar, are native just in Sahul, in New Guinea, in Indonesian West Papua and Papua provinces. In the other, in the rest of Indonesian territory, in Wallasa and Sundaland, there are no native crayfish. Crayfish in New Guinea are all of them are from the genus Herax. Indigenous crayfish in Indonesia, which are of New Guinean origin, included currently 26 species which were scientifically described, and 24 of them are strictly endemic. 2 of them, Herax quadriceratops and Herax rhynchotus, occur also in Australia. Here is a map of Indonesia and New Guinea, West Papua and Papua provinces. You can see that Herax gerardi is the first crayfish which was described by my team in cooperation with IPB and Dr. Daisy Bobor from the museum, the Zoology Museum in Bogor. And the second one is Herax snowden, junior synonym Herax subterigneus. And the third one is Herax acherontis. All of these crayfish are endemics of very restricted area, just one lake or one river. The important crayfish is the last one, Herax acherontis. At first, which is the total number of indigenous crayfish in New Guinea? It is unknown, of course, especially from PNG. There are no data about freshwater crayfish. And 26 species are described, but the estimation is up to 100 species. It's really high value, similar to Australian crayfish, and therefore we can call Papua or New Guinea, as you wish, as a biodiversity hotspot of freshwater crayfish. The first hotspot is North America, the second one is Australia, and New Guinea is expected to be the third one. The last one, Herax acherontis, as I told before, is a very important species because it is truly troglobitic. It's a cave crayfish. All cave crayfish inhabit flooded caves in North America, and no one was described before this finding in the southern hemisphere. All Australian astrobiologists would like to find some cave crayfish in Australia. They are searching for this crayfish more than 80 years and unsuccessful. Fortunately, me and my colleague found one cave crayfish in the Papua province, and it was really surprising for us and for other scientists that there is a cave crayfish in the southern hemisphere. The crayfish was found in the central highland in the vicinity of Wamena city in the Papua province. And therefore, we expected that more caves in this region can be inhabited by enigmatic crayfish as well. But the research in this region is really a hard job because we need scientific research permits and traveling permits and so on, and it is not so easy to go back to research the locality. But in future, we believe that more cave crayfish will be found in the Papua. New species, not cave crayfish, but epigeic crayfish are discovered year by year in New Guinea. Here are some examples: Herax pulcher, Herax barsamsonicus, and Herax alicae. You can see that all of these crayfish are very attractively colored, that their body and claws are colorful. And therefore, these crayfish are very popular as ornamental animals. These crayfish are exploited by locals for human consumption, of course. Here is a picture from river Aikima in Papua, and some local fishermen hunt crayfish for human consumption. But the most attractive species for human consumption is Herax monticula. And the others are, or many of them, are exploited for the international pet trade as ornamental creatures. Here is output from our analysis, because my country, the Czech Republic, is the most important or leading importer of ornamental crayfish in Europe. And Indonesia was identified to be the most important supplier of ornamental crayfish globally. You can see that more than 80% of ornamental crayfish are exported from Indonesia. And which species are exported? There are some Herax crayfish on the picture. On the right side, you can see one of the Herax crayfish, which are used for ornamental keeping. But surprisingly, just not only Herax crayfish, but also some others like Procambarus, the red one, which are non-native in Indonesia. These crayfish are native to North America, but they are exported from Indonesia. Therefore, we expected that they must be produced also locally in Indonesia, of course. And The research was focused also on this point, how the pet trade is working. The native crayfish from New Guinea are transported to the city of Sorong, and from Sorong by plane to Jakarta. In Jakarta, there are plenty wholesalers who are trading with ornamental animals in general, not only crayfish, but also fish, shrimps, and so on. From Jakarta, these animals, including crayfish, are transported abroad, exported to the United States, to the Europe, and into the Czech Republic, and to Japan. And the amount of transported crayfish is rising year by year, and therefore the pressure on native populations is higher and higher. And the overharvesting is a real threat for native crayfish in New Guinea, because many of these species have been previously not captured by locals, but currently the traders and the middlemen ask for these crayfish, and the demand on ornamental species is rising. Therefore, many local fishermen focused on these crayfish, and the overharvesting of native populations in their very restricted native range is a real threat. There are also some examples of non-indigenous, non-native crayfish in Indonesia, not in New Guinea, but in many islands like Java, Sumatra, Lombok, Kalimantan, Sulawesi, and so on. Here are these examples. The first one is Herax quadricarinatus. This crayfish, the red claw or red claw crayfish, is native to the northeastern Australia and southern New Guinea, but in the rest of Indonesia it is non-native, of course. And we found this crayfish in Java for first time in 2015, but the culture of this crayfish there is running from circa 2003. Many of cultured crayfish escaped from the aquaculture facilities and established new populations in the wild, feral populations. You can see that currently there are plenty localities where the crayfish established new populations. And there are produced just not only for human consumption, but also as ornamental animals. Here is example of one lake where crayfish are produced. Its name is Kemang Lake, and it is located in Java, close to the Bogor city. And the crayfish were cultured in the net cages inside the lake, but easily escaped from the cages, and currently there are very populated in the rest of the lake. Another example, and very risky example, is Procambarus clarki. Procambarus clarki, also called as red swamp crayfish, is native to North America, and it is known to be a very successful invader when introduced to some new localities, like in Europe, in Africa, in Japan, in South America. And this crayfish is exported from Indonesia in huge quantities. Therefore, we focused also on this species to find some localities where it is cultured. And we were successful. In 2017, we found this crayfish in one locality in Java. In its culture is running in outdoor ponds, and the crayfish can easily escape from the ponds and penetrate new localities in the vicinity. We found the crayfish there in huge quantities. You can see a video here that one catch of crayfish was successful, and we found many females with eggs there. And what is important, we detected for the first time in Indonesia a pathogen causing crayfish plague. The pathogen is named Aphanomyces astacei. It is fungi-like organism from the group of Olmeceta, and this disease is so risky to all crayfish of non-North American origin. The North American crayfish are vectors of this disease, and therefore the red swamp crayfish, Procambarus clarki, is a vector in Indonesia. What more? We found the infection also in two other decapod crustaceans in the locality. Paratelphusa convexa, which is a native crab there, and Macrobranchium lanchesteri, an invasive shrimp. Both are infected by the pathogen, but the concentration, the level of the infection is not so high, and therefore it is not possible to currently say if these crustaceans are sensitive to the disease or not. If yes, so the huge mortality can follow. But if not, both these crustaceans can serve as a vector of crayfish plague and spread the disease to the vicinity as well. So this is really bad news for Indonesian crustaceans and Indonesian crayfish. What is it? Crayfish plague. So the pathogen is Aphanomyces astacei, as I said before. Here you can see example from Europe. And this dead crayfish is Austropotamobius polypes, one of the indigenous crayfish in Europe. And all European crayfish are currently in risk because when the pathogen penetrated new localities, it is lethal for all native crayfish there. And therefore, this is a really serious threat for Indonesian crustaceans, not only for crayfish in New Guinea, but also for crayfish which are cultured in Indonesia, like Herax quadricarinatus. And we would like to do our best to mitigate the risk of the spread of the pathogen, but it is a really hard job because there are no effective restrictions and limitations in Indonesia to stop the culture of the vector of Procambarus clarki. So we will see in future if we will successful or not. The crayfish plague in Indonesia was detected for the first time in tropics, in some tropical region. And nobody can currently say what will follow because we have just data from the temperate zone, not from the tropics. Yeah, and here is a map of New Guinea. And there are plenty indigenous crayfish, as I said before. And because we found the crayfish plague in Indonesia, so we would like to predict what can follow when the pathogen and the invasive crayfish will penetrate New Guinean territory, and therefore we can say something about risks related with the indigenous crayfish if the pathogen and invasive crayfish are risky for this region or not so much. Based on the climate matching, we prepare the analysis. There are native crayfish, as I said before. Here are some examples again. And we made the analysis by some tool which can compare the native range and the target area of New Guinea in case of climatic conditions. And therefore, we can predict the probability of establishment of non-indigenous crayfish in the target region. We selected 5 very popular ornamental crayfish, which are non-indigenous to Indonesia, to New Guinea. 4 of them are cultured, in Indonesia, especially in Java and other islands as well. And one of them, the marbled crayfish, is currently not cultured in Indonesia, as is my knowledge, but it is very popular ornamental animal, and I assume that its culture in future in Indonesia is likely. The first one is Procambarus clarki. You can see that the red zones, which indicate a high probability to establish new populations in the target region. The probability is very high. Procambarus clarki is simply the most uh, risky crayfish from the collection to in perspective on indigenous crayfish in New Guinea. The second one is Procambarus virginalis, the marbled crayfish. As I said before, this crayfish is not currently cultured in Indonesia, but if in future this crayfish will penetrate Indonesian territory and New Guinea, so it is also risky. The third one is Cambarolus patsquarensis, dwarf crayfish from North America. Its total body length in adult stage is just 3 or 3.5 centimeters. It's a dwarf crayfish. But as because it is North American species, it can serve as a vector of crayfish plague as well. And it is also risky. Procambarus alani, Florida crayfish from the North America, from Florida, is not so risky as all of previously shown crayfish, because the climate matching indicated no similarity between the native range and the target area. So one can say that Procambarus alani has very low ability to establish new populations in the island. And the last one is Carax destructor. It is of Australian origin. This crayfish is very popular for human consumption and also for ornamental keeping. It is cultured in Java, and when penetrated New Guinea, so it is not so risky as, for example, Procambarus clarki, because the similarity of climatic conditions is not so high, and moreover, this crayfish is sensitive to the crayfish plague, so it is not so risky as North American crayfishes. But just only one, which is of a low risk, is Procambarus alani, of course. Here are examples of crayfish aquaculture facilities found in Java Island. The first farm, letter A, is close to Purwokerto. There are produced mostly Hyrax quadricarinatus, or just only Hyrax quadricarinatus, the crayfish from Australia and southern New Guinea, and they are used just for human consumption. The letter B is from Magellan, where crayfish are produced for both purposes, human consumption and also for the ornamental keeping. Letter C and letter D are examples of produced crayfish. Letter C is Hyrax quadricarinatus. Letter D, there are red swamp crayfish, Procambarus clarki. These crayfish are, the picture was captured and was taken in some pet shop in the city of Bogor. And some crayfish in the pet shops are positive on crayfish plague. It is not seen on them because they are resistant, but the pathogen is inactive on their body, and when they're mauled, their carapace or die, the pathogen will escape to the water and can infect new hosts. And about crayfish pathways, how they are transported within the Indonesian territory. We have just data from Java Island, and the monitoring will continue in future in other islands. You can see that the crayfish are transported from the Sorong to Jakarta, the yellow line from New Guinea to Java, and also from Australia to Surabaya. The import of Carex quadriceratops was for first time from Australia. It was surprising for us because we thought that the origin population was from Papua, but it's not true. The origin was from Australia, and they were transported to Surabaya, and there are plenty transport roads, as you can see the red lines, via which the crayfish are transported in Java from city to city. And there are many localities which are penetrated by escaped crayfish. And because the climate conditions in Java are very how to say suitable for many of these crayfish, so establishment of new populations is very probable. It is bad news for indigenous small decapod crustaceans and other invertebrates, and also for small fishes, because Heras quadriceratops, for example, can grow up to 20 centimeters, 25 centimeters in total body length, and it is also a predator. So it is not a vector of crayfish plague, but can outcompete native decapod crustaceans like small shrimps or small crabs and so on. So what to do? How to improve the situation? Easy way is total ban to North American crayfish, to the import of North American crayfish from abroad to Indonesia. It is a good plan, good idea, but some of these crayfish are currently present in Indonesia, and therefore the ban will be ineffective. Monitoring of indigenous crayfish species and monitoring of non-indigenous crayfish species is crucial. We need to improve our knowledge about these crustaceans in Indonesia to prepare effective approach, effective management of these animals, and try to help the indigenous species. Very important is also testing of sensitivity of other decapods like crabs and shrimps to the crayfish plague pathogen, because nothing is known about their sensitivity. There are just 2 species of European crabs which were tested, and some species of shrimps. But there are plenty species of decapod crustaceans in Indonesia, and nobody knows if they are sensitive or not. The 2 crabs in Europe, as I said before, are positive, are sensitive to the crayfish plague. Therefore, one can expect that more decapod crustaceans are sensitive as well. And if yes, so the crayfish plague pathogen spread in Indonesia is really bad news for indigenous or native animals. So testing of the sensitivity of other decapods to crayfish plague is crucial. What can we do relatively easily? We can say which species, pardon? Oh, which species are risky and which are of a low risk. Because we can replace the high risk species by low risk one. For example, Procambarus clarkii can be replaced by Carex quadriceratops. And further education of general public is also very important, because many of Indonesian fishermen and pet shop owner and so on has a poor knowledge about related risks about non-native crayfish and the crayfish plague pathogen. It's similar to Europe. Therefore, we decided to establish the Indonesian Crayfish Research Group, ICRG, which is led by Dr. Jon Wittner from IPB, You can see the logo here, and the activity is focused on both endemic and invasive freshwater crayfish, lobster air tawar, in Indonesia. We would like to evaluate the related risks and threats and the social economic impacts and so on. We would like to survey pet market, aquaculture. We would like to test the non-native diseases, distribution of crayfish, and new species discoveries is also very important. For this reason, we would like to start with popularization of our findings, not only in English and in scientific papers, but also in Bahasa Indonesia. We would like to start with the website, which is currently in development, and we would like to spread important information in Indonesia. We would like to discuss with local fishermen and our colleagues in universities and so on. And one note about Procambarus alani. As I said before, we have knowledge that this crayfish is exported from Indonesia in its blue coloration morph as ornamental animal, but nobody knows where this crayfish is currently cultured. Therefore, we would like to ask for the cooperation. If somebody has some useful data, some useful information about this crayfish culture, do not hesitate to ask me, to contact me, and I can offer a scientific publication as a reward of this cooperation. I'm open to the wide discussion and wide cooperation, which is currently running with several colleagues in Indonesia. So I would like to continue with the cooperation, and I'm ready to invite new colleagues to our team and start the cooperation. If somebody has the data about Procambarus alani, please contact me via email or WhatsApp and so on, and we can prepare the publication on this crayfish. So here is my email again. And that's all for now. Thank you for your attention, and I would like to start the discussion. If you have any questions, I'm ready to answer. Thank you so much. Terima kasih for your attention.
Terima kasih, Dr. Chiri Patoka. Thank you very much for the very interesting presentation. And before I start to let the participant to ask question, I know that Dr. Romi is also joining this seminar. Hello, Pak Romi. Okay, maybe later. I would like to give him opportunity to greet you, Dr. Chiri Patoka.
Yeah.
Yes. Okay, and then we have time for the discussion. So for the participant who would like to ask question to Dr. Chiri, you could unmute your microphone and ask the question directly, please.
Or you can write the questions to the chat if you want.
Yes, of course. And also you could write the question in the chat room, either in the Zoom or in this in the YouTube. So yes, there is two questions from Pak Hartoyo. In your opinion, which one better to do for Indonesian related to optimize of crayfish production as consumption product or as ornamental one? And the second is it potentially to make a massive culture for crayfish, especially for the consumption, human consumption?
Okay, so the point one is production of crayfish for human consumption and ornamental culture. I assume that there are plenty new Guinean crayfish which are so attractive as ornamental animals. They are native to Indonesia, and I would like to try to start the artificial production or captivity production of these animals. in Indonesia because they are endemic to Papua, and intensive capture can be very devastating for them. And if there is any possibility how to produce these crayfish in captivity, for example, in Java and so on, so this is the best way, I assume, how to produce ornamental crayfish in Indonesia. On the other hand, the production of North American crayfish is really risky to Indonesian biota, and I would like to do what we can to mitigate the production of these crayfish in Indonesia because it is really risky. So my suggestion is develop methods of culture of New Guinean Hyrax crayfish. It's the best way.
So the native crayfish from New Guinea is potentially be cultivated for only for ornamental fish, ornamental one, or for human consumption.
Yeah, so when the culture will be established and the methods of reproduction and rearing will be developed and established, so also the consumption is possible because these crayfish has meat of a high quality, so they can be consumed by humans, of course. But they are too aggressive, and the culture of these crayfish is currently not beneficial. And some new methods how to mitigate the aggressivity of these animals must be developed at first. But they are not so risky as North American crayfish. North American crayfish are easy to culture, but the risks related with the disease transmission and so on is very high.
Okay. Okay, thank you. And I think Pak Romi is already with us. Hello, Pak Romi.
Hello. Hello, everyone.
Please greet to Dr. Chiri.
Hey, Jiri, how are you?
Hey, hello, Romy. Nice to see you, or just virtually again.
Okay, can I ask question?
Of course.
Yeah.
Please.
Yeah. On your first slide, you told us about the biodiversity hotspot of these things, yeah. And I saw most of line is actually in line with the Wallace line, but you have the second line is close to Papua, which is third hotspots appear there. It's also I believe some other group of organism also having the similar pattern. What on this case? What causing the third hotspot appear in that area, especially in the Papuan?
So I'm not sure if I understood the question. So the crayfish are native just in Sahul, not in Wolosa and Sundaland. So the richness of this group of crustaceans is just from Papua. Or what was the question?
Yes, I saw the three hotspot there, and then you saw that one native to Sahul, right?
Yeah. Okay. So the hotspots are general, so biodiversity hotspots, not only of crayfish, and crayfish are just a part of Sahul.
Okay, thank you.
Yeah. Okay. So I'm not sure if I understood. Sorry. So maybe you can ask me again via chat.
That's okay.
Okay. So
thank you.
And Romy, wait.
Yeah.
So I have a good news for you to you because our paper on marine petrite in Indonesia was accepted for publication in Aquatic Living Resources.
Okay. Good to hear that.
Yeah. So I sent you an email with this information, but I'm not sure if it was delivered. Maybe can you contact me via email again, because I'm not sure if we are in contact.
Yeah, I quite seldom to open that email. I use the other email.
Yeah,
I will give you
perfect.
that email. So this is the second of our paper, right?
Yes, yes, yes. The second one. So thank you very much for such a perfect cooperation, and I would like to continue in future.
Yes, indeed.
Yeah, thanks.
Okay,
And
thank you very much,
hopefully see you next year in Indonesia when the COVID pandemic will be terminated. So I would like to go personally to Purwokerto again.
Yeah, the program is only postponed, so welcome next.
Thank you,
Okay,
Jerry.
thank you so much.
Thank you.
Okay, thank you very
Nice.
much, Dr. Romi and Dr. Chiri Patoka. It is quite nice discussion, and I hope that the species Procamborus aleni could be found in Indonesia. I mean I think you are needing the location of the cultivation of this species.
Yeah.
Yes.
So I assume that Procamborus alani will be cultured in the surrounding of Jogja. There is the most probable place where the crayfish is cultured because there is the crayfish aquaculture well established in Jogja. So maybe this crayfish also is cultured there. But I have no data. If somebody have some information about this crayfish, it can be very beneficial for all of us.
Okay, thank you very much, Dr. Chiri Patoka. And because limit of time, we have to finish our discussion. Once again, really thank you very much for your presentation and discussion. So please, all participants, we give great applause for Dr. Chiri Patoka.
Thank you, Jerry.
Thank you so much.
Thank you.
Sama-sama.
Terima kasih.
Terima kasih, Jerry.
Sama-sama. Thank you, Pak Amron, for inviting me to this seminar. Thank you.
See you.
See you later, probably next year, I hope. Yeah.
Okay, see you, Dr. Chiri. And now, for the third session, we already have here Dr. Mareike Hun from Germany. How are you?
Hi, good morning.
Is my voice clear enough from your side?
Yes, yeah.
Yes, let me introduce myself. My name is Hamdan Sakuri, and I'm the moderator of this seminar today. Yeah.
Good morning, atau selamat siang ke Indonesia.
Selamat siang.
Selamat pagi waktu Jerman, mereka.
Guten Morgen.
Masih pagi-pagi.
Masih pagi-pagi.
Entschuldigung, das ist zu früh. Ich denke.
Oh, saya tidak mengerti.
Wie spät ist es in Deutschland?
7:45 Uhr.
7. Okay.
Jam 7:45.
Okay, ladies and gentlemen, let me introduce our third speaker. Her name is Mareike Hun.
Yeah.
Yeah, Mareike Hun.
Yeah, Mareike Hun.
Yeah, Mareike Hun. Currently, she is postdoctoral researcher in Ruhr University Bochum, Germany. And Dr. Mareike has quite a lot of experience with Indonesia because I think since 2011, she was in Indonesia for 6-month research at Bogor Agricultural University for her master degree. Yes, it is for the master degree. And then He also has collaboration again with Bogor Agricultural University between 2012 until 2006. And interesting enough that since 2018 until present, he also director of Luminocean, PT Marina Citra Selaras. So actually, she could speak Indonesian excellently, and also, of course, English. But bitte nicht Deutsch sprechen. Okay, I think you could try to prepare your slide.
Yes.
So please, the committee allow her to display the slide. Yes, it's working. So Dr. Mareike Hoon will be presenting her paper about Ascidian as bioindicator. I do not know quite a lot about the Ascidian, but I know that it is a group of invertebrates, marine invertebrates, I think. And for today, you have 30 minutes for the presentation, and then we will have another 10 minutes for the discussion. So for the participants, please, during her presentation, you could write your question directly in the chat of the Zoom meeting or in the YouTube. So Dr. Mareike Hoon.
Social enterprise to strengthen higher education, especially in East Indonesia, in the Moluccas at the Banda Islands. So we offer student scholarships to local students and mentor them so that they can do practical research associated to diving. So yeah, maybe some of the students might come across Luminocean later on in their career. But today I would like to talk about Ascidians because first time I came across them, I started working with them was through a bioinvasion monitoring that I did together with IPB. And now I just started another project with a Mediterranean species of Ascidians. And I think, both are quite interesting. Ascidians belong to the tunicates. So this is an order that comprises 3 classes, the Thaliacea, Appendicularia, and the Ascidians. And within the Thaliacea, there are 3 subclasses. One are the tiny doliolids. They're just 1 to 2 millimeter long barrel-shaped animals, and they live in the pelagic. There are also the salps. These are colony-forming species, pelagics as well, and they form long chains, gelatinous. And if you sometimes go diving or snorkeling, maybe you have seen them in Indonesia. I have seen them in the Banda Islands. And also pyrosomes. They are also colonial pelagic tunicates. And what is fascinating about them is that they can form colonies up to 3-4 meters long. A person would completely fit inside them. And some of them are even bioluminescent. The second class are the Appendicularia, or they're also called lavaceans, because they kind of remain in a kind of larva stage even during their adult life. So what you see here, that is what is a typical tunicate larvae. And what is special about the Appendicularians is that they remain in this stage and they just form this gelatinous housing.
and this helps them to filter even tiniest particles from the sea, so they can feed from bacteria, from microorganisms, and have a quite special role as filter feeding pelagic organisms. And then the last class are the Ascidians. This is probably what everyone is most familiar with. They are benthic, mainly benthic invertebrates. So you find them attached to hard substrates, and they also have quite fascinating lifestyles. So in this talk, I will focus on this group. Ascidians are barrel-shaped animals. It's hard to imagine that they're animals when you see them settling on a rock, but yeah, they are. And they look very simple, look similar to sponges, but the biggest difference to sponges is that they have two siphons. So you can see here they have an oral siphon where they suck water in, and the water flows through the branchial baskets, where that retains all food particles, but it also is responsible for oxygen exchange. And then the water is pumped out again through the atrial siphon, where it releases all the substances that it cannot use, and also the deoxygenized water. Ascidians have a tube-shaped heart, and you can see here, this is the heart, which is pumping their hemolymph, or their blood, basically. And this heart can pump backwards and forwards. So I'll play this video again. So sometimes it flows in this direction, but they can also reverse the blood flow. It's not exactly known yet why they can do it, but it's assumed that this is responsible for that they can better provide blood to all their organs or their body parts. And Ascidians can be either colonial or solitary. The one on the top left is a colonial Ascidian, so you can see it looks, from far away, it looks completely different to the normal shape of an Ascidian. So here there are thousands of tiny zooids that are covered by a common tunic. And the tunic is the mantle of the Ascidian. It's a leathery skin, basically a leathery mantle that covers the whole animal. So here in the colony, the mantle is the white part covering all the zooids. So one individual inside the colony is called zooid. And here the mantle is the purple and blue part. So it can be also quite transparent. And this is another beautiful example of a colonial Ascidian. So sometimes they are not like here, just looking like one white, or can be red, or any color basically cover. They can also look like a beautiful patterned colony. And here you can better see each individual zooid with two siphons. So this would be oral siphon, and this an atrial siphon. So in this colony, each individual has two siphons to take water in and release water again. But in other species, they can also share the atrial siphon. And this is an example here. Here again, you have the typical shape of a zooid, and it has its own oral siphon, but then it shares an atrial siphon with other members of its colony. And then you can imagine that the mantle, the tunic, is covering the whole colony. The reproductive system is also very interesting. In colonial Ascidians like this, they can alternate between sexual and asexual reproduction. During sexual reproduction, the adult animal releases eggs and sperms. So they are all hermaphrodites. They can release both either eggs or sperms, but there are mechanisms that avoid self-fertilization. So they would always mix with a different zooid, not fertilize their eggs themselves. And then the eggs develop into a larvae. The larva stage is quite short compared to corals, for example. Corals have larvae that spend 3 weeks on average, I think, in plankton. And for Ascidians, this can just be 20 minutes in some cases. Then they directly settle and develop into a juvenile animal. But the colonial Ascidians can also conduct asexual reproduction, where they just form buds from all kinds of different body parts. So this can be from the stolons. Stolons, you can imagine like the roots that they use to anchor themselves to the substrate. But it can also be from the tunic, and across the different taxa you can find budding in almost any body part of the Ascidian. In contrast to the colonial Ascidians, the solitary Ascidians only reproduce sexually. So they're also all hermaphrodites, and so they all release eggs and sperm, but never self-fertilize them. And then they develop into this free-swimming larvae, which looks like a tadpole larvae, like a tadpole, or almost a little bit like a little fish. And only when you look at this larvae, you can actually see their evolutionary origin. So these invertebrates are basically the link between chordates and non-chordates. So they are our closest relatives among all the sessile animals in the sea. And you can see this in the notochord and the nerve cord that this larvae has. So they're also very interesting for neurobiologists for studying the evolution of the nervous system, because you can see evolution happening in real time here, basically. And this larvae has a cerebral vesicle. So this is like a tiny brain, basically, a place where the nerves concentrate, and it can perceive light and dark. And it also knows where perceive gravity. So it knows where it's up and down. And this is all the information it needs to start its adult lifestyle. So it finds the suitable place to settle. And when it settles, it goes upside down and anchors itself with its head, and then it starts to digest its tail, because everything that it needed for swimming around this tail, it doesn't need anymore. So it starts to digest it. It doesn't need to feed until it transformed into a juvenile, and it can start filter feeding. So this for especially for tropical species, this can happen, larva stage within 20 minutes, and then just over several days to weeks until the juvenile has formed. As I said, it's a really good model for neurobiologists, because these larvae are transparent, so you can really easily visualize action potentials, visualize the nervous system with fluorescent staining, for example. Ascidians are really widely distributed, and I found this map really interesting. The numbers that you see are the species described, species of Ascidians described. And you can see that generally there's a higher abundance in the tropics, especially in the Indo-Pacific. But what is quite striking is that Australia has described 717 species, whereas in Indonesia there are only 57 species described so far. And I'm sure in Indonesia there are many, many more. I think that this outstanding number for Australia is mainly because they do a lot of taxonomic work, also genetic taxonomic work. And when we did work at the Banda Islands and collected samples of Ascidian and sent them out for barcoding, we found hardly any matches in gene banks. So I'm sure there are many still undescribed species in Indonesia, which is, I think this could be a really interesting field of study for students who still want to decide what they want to focus on. There's a huge potential here. Ascidians, because many of them have a very opportunistic lifestyle, they just need plankton to feed on. That's why they also have become invasive species, or some of the species have become popular invasive species in the world that can also cause economic problems, like some Didymnum species, especially Didymnum vexillum or Didymnum pellucidum. And you can see in the left picture how Didymnum vexillum settles on oyster cages. These are cages in oyster farms. And this Ascidian grows so quickly, excuse me, that it makes it hard for the oysters to still get fresh water supply in their cages. And they are transported especially on boat hulls as fouling or through aquaculture. And yeah, knowing this, that in other parts of the world, Ascidians can create troubles, we started, so we, that is researchers from Bogor, Hafiz Madupa, and some students from Bogor, together with Australian researchers, we conducted a bioinvasion monitoring at the Banda Islands in 2015, 16, 17. And this was funded by Australian National University. They have a special grant for research projects from Indonesian researchers. And what we thought was, let's see what fouling organisms are there in the Banda Islands that are not corals, because corals are very well described, but the rest is often under-described. And we put out these settlement panels to collect spat, so larvae of any marine invertebrates, and we're monitoring them for 2 years. So this is also published in the journal Biological Invasions. I'm not going to talk in much more detail about this project, but what was really interesting from this project was that we found one Ascidian species called Didymnum molle, which was strikingly covering all the many panels that we put out, and especially also the ropes of the panels. And yeah, later on, we observed them also growing on corals and covering a really large area in the reef in the Banda Islands. And if you ask, or if you try to search information, what is the native range of the species? So where does it originally come from? Where did it possibly invade? Then you just come up with a huge native range. So everything you see in red here it's to date considered native range. And it's so huge that from this you can tell that no one really knows where it actually came from. But the predicted ranges until 2050 are that they might spread also to other tropical areas. And what I think is that of a species, it's not always the problem, is it native or not, but the question is, what does it cause in a reef if it starts to grow too much? So later on, yeah, students from the Fisheries school in Banda and Herdian Wicaksono, who actually graduated in Purwokerto, maybe some of you know him, they continued working on this topic in the Banda Islands to monitor how the species spreads, or does it spread, and what are the correlations with a very high abundance in some reefs and a lower abundance in other reefs. Because we thought, okay, we cannot say is it native or invasive, but we can definitely see that it's starting to take over a lot of space in the reefs. So the abundance of the Demnomole was quantified in several reefs. So these are the Banda Islands. It's a volcanic group of islands in the Moluccas. From, I'm sure all of you know Ambon. From Ambon you drive by boat 12 hours southeast into the sea. It's quite far offshore, very remote, but beautiful place. And this is the central group here, the volcano island, and the most populated island. And then there are a few smaller, more remote islands, about 10 kilometers outside. So what they did was to put out transects in the reef in 4 different depths, 6 to 18 meters. And at every 1 meter mark, there was a frame placed 20 times 20 centimeters that was photographed with a camera. And then from these photos, the percent cover of the Demnomole was analyzed. And then it was seen like how does the abundance of the Demnomole relate to different types of substrates. So these are substrate types typically used by Reef Check for Reef Check surveys. And what you can see is that they preferred rock coralline algae, hard corals, and also sponges as substrates. So, and especially the hard corals were surprising. So these are the substrates where they dominated. And then looking at the substrates where they were most dominant at the different sites, you can see that there's a huge variation between sites. So this is where basically we started to see what is the reason that they occur in large numbers in some sites, but in low numbers in others. And strikingly was that the sites with a high abundance were those that were either really close to the natural harbor, more to the central islands that are more populated, where you have more anthropogenic impacts, and also a reef number 3 here, where you find a lot of damage by anchors in the coral reef. So there's more dead coral available for the Ascidians to settle. But it was overall a very variable data. So what Ascidian did then was to check whether, when looking at the coast of east coast of Banda Besar, biggest islands of the Banda Islands, where you have very similar reefs all along, looking at what is the pattern there, how are the Ascidians distributed. Because we thought maybe if they start spreading from the inner islands, you would find a gradient that you find most here, and then getting more to the remoter areas, you would find less Ascidians. But this was not really the case. So these are the different sites. From number 4 is this, and 4.55, the last one. And you can see that you have areas where there are not many, and then suddenly they pop up again, basically. And most of them were found at 10 and 14 meters, which is at the Banda Islands, the reef crest, and just below the reef crest, so where the slope starts to become steeper, or the upper meters of the wall. So this is probably where they have a little bit less competition by hard corals, but still enough light available, because same as corals, they also have symbionts. All the green spots here, it's prochloron. That's a symbiont that also conducts photosynthesis and provides them with sugars. Then there was another study done to see how quick they grow or disappear again from different substrates. I'm going a bit quicker now because of time. And it was compared how they grow on hard coral, dead coral, and rock. And what was found here was that the biggest changes occurred on dead corals. So after 60, 120, and 180 days, always photographing the exact same colonies on a predefined frame, the biggest fluctuations occurred on on dead corals compared to rock and hard coral. So this is the beginning of a study, I think, that can still develop into much, much more, because there's still a lot that is not understood yet. But what is understood so far is that they prefer to settle on dead corals, also like rock coralline algae, and also especially the same habitats as corals. And the abundance, however, fluctuates highly, which makes it really difficult to study. And you can see, if you look at them with time-lapse camera, you can see a lot of variation in colony spread and abundance. And the predation is very low. We never saw any predation, I think only in 2 cases there was a predation event, and these are normally tiny crustaceans that live inside them, or sometimes snails. So this is an interesting species that, if more is known about it, I think can be used as bioindicator of reef health, because it always starts to grow, comes up when a reef is damaged, especially if hard corals are damaged. What will be interesting now after coral bleaching this year, if they can be seen to develop more on the reefs. What is not known yet is if they will have a long-lasting impact. Maybe they just disappear again and won't have a bad impact, but maybe they do. It's known from lab studies that they produce substances that inhibit growth in corals. So then I come to the current project now. So now I'm looking at a solitary Ascidian, so completely different, similar but different, from the Mediterranean Sea. And this is Halosynthia papillosa. It is only found in the Mediterranean Sea and the Northeast Atlantic, so it's not as widely distributed as the Didymnomolle. Also settles on hard substrates and has an extremely tough and rigid tunic. This tunic is 2 to 3 millimeters thick. And it gets quite big, 10 to 20 centimeters, but also feeds on tiniest phytoplankton. And interesting about it is that it's considered as bioindicator for too much scuba diving. So this was a study by a Spanish team, Luna Perez et al. And they found that there's always a correlation that these species disappear after an extensive scuba diving season, but in marine parks where there's no scuba diving, they don't disappear. So they simulated what divers do with their fins if they paddle too much and stir up too much sand. Then the tunicates get covered by sand, and they found that they are disappearing in the treatments with a lot of sand. So here you can see, you can look at the different treatments. So with the highest fin flaps, that's number E, just after 24 hours, they started dying. After 1 week, a lot of individuals were dying, whereas in the control without sand, none died over the whole time. So this is the total number. The other lines are different sizes. So the more fin flaps they got on one event, the faster they died. So I think this is quite interesting, and maybe it can also be applied in Indonesia to find out whether reefs are under stress from scuba diving by finding a suitable comparable Ascidian species, because you normally see a reaction in Ascidians much faster than a reaction in hard corals. So they might tell you already before corals tell you that there's a problem in the reef. So we went to the Mediterranean Sea and to Croatia and collected the species and brought it to the lab in Bochum. And now I'm running experiments with them to check their tolerance to thermal stress to see how they would do in future climate change scenarios. And especially I'm also testing which response variables work well to find out if an Ascidian is under stress before monitoring mortality. So I'm focusing especially on heat shock proteins, because these are proteins that protect the species when they're upregulated from thermal stress, but also from other stressors. So if you find the right heat shock protein, it's a whole protein family that is produced in any organism basically, you can find which ones are suitable for which stressors. So the first experiment I did was exposing them to an increasing thermal stress, one heating permanently with 1 degree per 30 minutes and one with 1 degree for one group with 1 degree for 1 per hour, to see what their limit is, what their thermal limit is, and to isolate proteins to see how they are producing heat shock proteins. And they, even though they are a Mediterranean species where you can see normally their ambient temperature is 10 minutes to 10 degrees to 20, maybe sometimes they encounter 22 degrees, they survived until 31 degrees. That was very interesting. So the red group was the one that was heated quickly. They survived less than 400 minutes and died at 31 degrees in average. Here you can see the temperature. This is the temperature curve that I had in the respective treatment. And the group that was heated slower also died at the same temperature, even though they had more time to acclimate. So I'm also looking at their reaction, and if you tap their siphons, you can see quite well how fast they're closing or opening. And I'm developing a fitness score so that this can even be done underwater while diving. If you tap, but you would most likely have to do this several times, tap an individual and then see how's the reaction and how is it performing. So this can be an indicator for stress level also. So here the control group in blue was more often responding quickly or having the body shape like fully blown and not contracted than the heated groups. This is a time-lapse video of how they can contract and expand. So when they're disturbed, they contract. And these were the first results doing Western blots for heat shock proteins. I still have to do much more, run more trials. I'm just still in the middle of it. But what we can see already is that the group that was slowly exposed to temperature produced an inducible form of heat shock protein 70. So this is actually a suitable heat shock protein that can be used as indicator for thermal stress. And interestingly, the group that was also exposed to heat but didn't have time to acclimate for a long time expressed way less of heat shock protein, of the induced form of heat shock protein 70. And here you can see this is the upper band here is a form that is always produced, and the lower band is an induced form. But I'm still working on optimizing these plots so that they can properly be analyzed. And now I'm running an experiment with a long-term moderate stress that is realistic for climate change scenarios. So yeah, I think what maybe you could take home from this, or especially as students, I hope you might get interested in this species because there's really a lot to do. It's a fascinating species group. There's a lot to do, especially in Indonesia, I think. And I think they might have potentials as bioindicators that haven't really been looked at that much yet, and especially for managing marine parks. Yeah, looking at the condition of coral reefs, using these as bioindicators would be a really interesting story. So yeah, some ideas maybe what to do. Are there, I'm sure we can ask many and not any unidentified species of Ascidians in the Indonesian archipelago. Barcoding will help a lot for this. And maybe can species in Indonesia be identified that can be used as bioindicators for too much scuba diving, but maybe also for sedimentation stress that often comes if there's dredging, if there's too much work on land. Which characteristics can be used for each species or for each stressor? And I'm sure it's easy to find indicators that are where you don't have to look at mortality of the species. And maybe can even, based on this data, local regulations be established, so this can contribute to coral reef conservation. That's my message for you for today, and I thank you very much for listening and attending, and of course I'm happy to receive questions. Terima kasih.
Terima kasih Dr. Marai Kehun. And then now we have 10 minutes for the discussion. So for the participant who would like to ask question, you could type the question in the chat room, or you could unmute your microphone and directly ask the question, please.
Kalau mau bahasa Indonesia bisa juga.
Oh ya.
Atau ini seminar Inggris.
Tapi bahasa Indonesianya sangat bagus ya. Silahkan para peserta. We could wait for the question from the participant, and then I would like to ask a question. Could you explain more about the use of Ascidian as bioindicator of coral reef health?
So the two examples I gave, basically, I think are two examples of how it can be used. One would be of monitoring abundance of any species that is very common, like the case of the Didymnomolle. So if you look at, if you go into a reef and you look at dead corals, if you look at spaces that just became free because a coral died, and you find that there's Ascidians growing, then this could be a suitable species to keep an eye on. And what would need to be done is then regular surveys to see if the species is becoming more or getting less, and especially also if you can observe them growing on living corals, because then they might really become a problem in the reef if coral destruction continues. So, and the other option would be, which is easier with a solitary Ascidian, to keep an eye on the abundance of this Ascidian, and if it disappears, that could be a sign that the reef is under stress. And because they're quite sensitive, they're opportunistic, but many species are also quite sensitive, they would probably suffer earlier from sedimentation stress, from temperature. Probably it depends highly on species, but I think if a little bit more work is done on temperature tolerance or other stressors tolerance, these could also be indicators of how will the corals react a little bit later on. And I think the example with the scuba diving is very good because it uses sedimentation stress, which is a stressor that is very bad for corals, but it seems even worse for Ascidians. Corals would not die after less than a week if you would throw sand on them once, basically. But if it's a repeated thing, then corals would also die. So when looking at the Ascidian and you see this disappears, then it's probably still early enough to do something to stop what is causing the stress, especially in the case of sedimentation.
Okay, thank you very much. And there is a question from Dia Ruri in the chatting room related to finding the protein family to reduce the heat shock protein. How to reduce their stress by using the switch protein family.
How to reach the stress?
To Dia Ruri, I think it is better to ask your question directly. Please unmute your microphone.
Yes, thank you very much, Pak Hamdan. It's a very nice material that you've presented today, Dr. Marika. So, yes, you're welcome. I recall that you mentioned related to the heat shock protein, and you say that it would be better if you could find the family protein that can switch to reduce the stress of the Ascidian. So how you possibly implement that to Ascidian in order to make them less stressful? Thank you.
The heat shock proteins, they're basically more a method to detect on which stress level they are. So you can't really apply them to reduce stress, but you can monitor how is the stress, and then you can try to change something that there's not so much stress anymore.
All right. So change something here means their habitat or their environment, right?
In the environment. So one example would be that you stop whoever is causing the sedimentation. You would stop them from continuously putting sediments into the sea, or from very good example is freshwater influx, which often comes through cooling water at industries. And salinity stress also triggers these heat shock proteins. So that could be another thing.
Okay, thank you very much.
And what I'm trying now is to get enough tissue from these Ascidians to measure heat shock proteins, but that they don't have to die yet. And if this works, could go just with a scalpel underwater, take a little bit of tissue, and then see under which stress level they are at.
Okay,
for colonial ones, it's easy because you can take a part of the colony, but for solitary ones, it's a bit more difficult, but it would be a nice thing. Of course, it needs to be established for each species because you have to see what is a high level of heat shock protein for them, what is a low level, and when can you say, this is now under stress.
So the HSP70 is used for stress indicator.
Yes.
Of the animals. So, and You do this in the level of the proteins. How about the mRNA?
Yeah, it also works. You can do real-time PCR for quantifying expression of these proteins. You can do Western blots with, that's what I'm doing at the moment. It's very easy, very robust, basically, but both works really well. And there's lots of studies done with both techniques already. On Ascidians, not that much, just on a few species that are common lab species, but on other species, marine species, there's a lot done already.
But I think you are right that the challenging thing is how to take the samples to not kill the animals, because it is, I think, tiny animals. Right?
Yeah, but they have a huge capacity for recovery. You can, even a solitary one, you can basically cut it in half and it will regrow the rest of its body.
Really?
Yeah.
It's interesting.
So I'm trying this now, how much tissue I need. So of course I want to take only as little as possible, that they recover well and it works.
Okay. Okay, Dr. Mareike Hun, because now it is in Indonesia 2:30 p.m. And I think Sie müssen auch Frühstück essen.
Ja, genau.
Right? And we are really sorry because yes, it is winter in Germany, so 7:30 is, I think, too early in the morning.
-2 outside.
Oh, really? Yes. And now, because limitation of time, we have to finish our discussion. Once again, thank you very much for Dr. Mareike Hun for presenting her work on Ascidians. And for all participants, please our applause to Dr. Mareike Hun.
Thank you very much.
Thank
You're
you.
welcome.
Thank you, Marika.
Thank you, Dr. Marika.
See you later.
Okay, it's nice to meet you.
Nice to meet you.
Pak Hamdan, maybe we can take a picture.
Okay, yes. I'm sorry, we have to take a picture. So please, the committee organize this. So everyone who could possible to activate the camera. One, two, three, four. Enough, Ibu Ruri?
Yes, suppose it will be all.
Okay. Once again, thank you very much, Dr. Mareike.
Thank you.
Thank you, Marika.
And ladies and gentlemen, it is the end of our seminar today. And I would like to thanks very much for listening to this seminar. And I give back the chance to Ibu Diah Ruri. Thank you.
Thank you very much, Pak Hamdan, for guiding the session. And thank you very much, Dr. Veronica, for delivering her expertise today in S2FiMaS. So, distinguished guests, ladies and gentlemen, now we've come to the end of our fourth seminar series today. We would like to express our immense gratitude to all invited speakers, moderators, presenters, and also participants who have joined the fourth series of S2FiMaS. We also would like to apologize to the inconvenience that we may We hope that you have a pleasant seminar and fruitful discussion. See you on the next 5th seminar series of S2FiMaS on the 17th of December 2020. I suppose it will be all. Thank you very much. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh.